Okay, good morning everybody. I think it's turned 10 o'clock, or 10 seconds to 10, so I'll let everybody sit down. So welcome to the symposium. My name is Mike Hanna. I'm a neurologist here at Queen Square, and I think I'm partly here because 20 years ago, in 2006, with Doug Turnbull and Joe Poulton from Oxford, we set up this NHS England highly specialised commissioned service for mitochondrial diseases, which many of you are linked to or will know about. And a lot of what we'll talk about today does link to knowledge we've gained over those 20 years in that service, which I think has become a useful clinical or diagnostic service for patients all over the country. What's central to that service, and I think will come out during the different presentations today, is the critical nature of MDT working in trying to work out an accurate diagnosis for patients and then working out what the best way to help them is. And I can't stress enough how important that MDT working is with the entire team meeting. It used to be physical, since COVID it's mainly virtual, I think. But that's neurologists, paediatricians, physios, OTs, pathologists, geneticists, biochemists, and others all working together to try and focus on the patient. And that will come across, I think, in the different presentations today. Also, I think the highly specialised service has enabled lots of research to happen and has enabled a very effective linkage between research colleagues and NHS colleagues, whether that's in genetics or pathology or in allied healthcare professionals, and I think that's really important. And of course there's important links with our charity partners, and I'm very pleased that the Lilly Foundation, the main mitochondrial charity, is here today. So just to give you a quick overview, we want this to be clinically useful. We're going to talk about different areas that we've listed here. So we'll touch on preclinical models and their relevance to translational research and clinical practice, update on primary mitochondrial diseases, the molecular diagnosis, biochemical diagnosis, and also consider the patient diagnostic journey, which we're trying to make quicker so that we get the right answer sooner. Historically, as you all may well know, it was often a very long journey of uncertainty to get to an accurate genetic diagnosis. And I'm very pleased that Bobby McFarland is here, and we'll talk about major advances in our ability to undertake prenatal genetic diagnosis, counselling, different preventative strategies, including mitochondrial donation, and also we'll touch on therapeutics as well. The other reason I know that I'm getting older is because I get asked to put this slide up on housekeeping. The symposium will be recorded. It will be uploaded onto a YouTube channel. Sign in at registration if you want an attendance CPD. We don't plan to have a fire alarm, so if there is one, leave the room. There's Wi-Fi as indicated here. We've just paid for a brand new lecture theatre, which I hope you like, which hopefully we don't want you to bring food and drink in. And UCB are also supporting this event. So I hope it's a really useful and interesting day for you. Hopefully you'll be able to interact a lot, ask questions, and interact at the lunch break and the other breaks. It's always good to try and talk to someone you've never met before, so try and make a point of doing that during the course of the day. Great. So I'm going to hand over now, I think, to Rob, who's chairing the first session, is going to do the first talk on primary mitochondrial disease.
Thanks, Mike, and welcome everybody. It's great to see so many people here. So I'm going to... my name's Rob Pitceathly. I'm a neurologist here at the National Hospital for Neurology, and I co-lead the NHS England highly specialised service for mitochondrial disorders with Mike here in London. So I'm going to provide a relatively brief introduction to primary mitochondrial diseases. I'm going to try and avoid treading on other people's toes, because you do have talks in other sessions that cover many important aspects, particularly around genetics and biomarkers and treatments. But I'm going to talk a little bit about mitochondria themselves, mitochondrial DNA, and primary mitochondrial diseases. I'm going to give a brief overview about some very specific concepts around mitochondrial DNA, which are unique to that particular genome versus the nuclear genome. And then I'm going to finish by talking a little bit about clinical syndromes, which when I reach that point, you'll see that we're moving slightly away from, given the more genomics first approach that we're taking to not just mitochondrial diseases, but all genetic diseases. So this is a mitochondrion. This is how they're often depicted in textbooks, although it's not really how they are in real life. So they're incredibly dynamic organelles. They form this sort of reticular network within cells, and they're constantly dividing and fusing, exchanging information, genetic information, according to the metabolic demands of the cell, increasing in number and reducing in number. But this is quite useful just to understand their basic structure. So they've got two membranes, an outer mitochondrial membrane and an inner mitochondrial membrane, which is highly convoluted. And the reason for that is it increases the surface area in order for all these very important metabolic reactions to take place. And there are lots and lots of those. But arguably one of the most important is oxidative phosphorylation, so the production of energy, or ATP, for the cell. And so that's what's happening here, and I've also shown the Krebs cycle as well. This is where this is a mitochondrial genome within the mitochondria itself. And there are lots of different pathways that can be implicated in diseases, so primary mitochondrial diseases, but arguably those that affect oxidative phosphorylation and those that affect the mitochondrial genome, particularly the way that this genome expresses proteins and the way that they're translated. Those are one of the two major groups of primary mitochondrial diseases. But that definition of primary mitochondrial diseases is constantly expanding as we identify new genes. And because mitochondria are so important for cellular metabolism, they've been implicated in all sorts of different diseases, some of which they're obviously a primary driver, some maybe just that we see problems with mitochondria as a downstream consequence of the underlying pathology. But what we're focusing on in today's symposium are primary mitochondrial diseases. See, these are genetic, monogenic diseases that affect the ability of mitochondria to produce energy, or the structure or the function of mitochondria. And as I say, that definition of what are primary mitochondrial diseases is currently under review as we identify more and more genes, some of which are arguably more peripheral to oxidative phosphorylation. So what is oxidative phosphorylation? Well, this is shown here. So this is the inner mitochondrial membrane. So that's that highly convoluted membrane. And there are a series of enzyme complexes that are embedded within this inner mitochondrial membrane, 5 in total, involved in oxidative phosphorylation, and they're multimeric. So they're comprised of lots and lots of subunits. For instance, complex 1, so then labeled complex 1, 2, 3, 4, and 5. Complex 1 is over 40 different subunits, all encoded by different genes, some of which are encoded by the mitochondrial genome, some by the nuclear genome. And essentially what these enzymes are doing is they're involved in a series of redox reactions. At the start, electrons are donated from food, and those electrons are then passed down these complexes in a series of redox reactions. And in doing so, they provide energy to enable protons within the mitochondrial matrix on the inner side of the inner mitochondrial membrane to be transported across into the intermembrane space, so that space between the outer and the inner mitochondrial membrane. And what that does is it sets up an electrochemical gradient across the inner mitochondrial membrane, which is harnessed by the final or terminal enzyme in oxidative phosphorylation, complex 5, and those hydrogen ions flow back across that electrochemical gradient into the mitochondrial matrix, and in doing so, they enable ATP to be produced from ADP and inorganic phosphate. And that's this process is the main source of ATP for the cell. So the other important thing to know about mitochondria, and I've touched on this a little bit already, is they contain their own circular genome or mitochondrial DNA. So every cell in the body has hundreds to thousands of mitochondria according to the metabolic demand of the cell. And that number of those mitochondria is constantly fluctuating according to how much energy that cell requires at any given moment. Each mitochondrion can contain multiple copies of this circular genome. And this is similar to nuclear DNA, but obviously it's a closed circle. It comprises around 16,500 base pairs, so only 37 genes. They encode 37 genes, and 13 of those 37 genes are subunits of these different enzyme complexes, apart from complex 2, which is fully nuclear encoded. All the complexes 1, 3, 4, and 5 contain subunits encoded by the mitochondrial genome, these 13 polypeptide subunits. The remaining 24 genes encoded by mitochondrial DNA are involved in synthesis of those. So they're ribosomal and transfer RNAs involved in intramitochondrial protein synthesis of these 13 polypeptide subunits. And given this circular genome is DNA, it can mutate, and the first pathogenic variants within mitochondrial DNA were discovered here at the Institute of Neurology by Ian Holt, Anita Harding, and John Morgan Hughes. And what they did for the first time was they extracted mitochondrial DNA from muscle tissue rather than blood. And they then, and this was in patients with a condition which I'll speak on a little later called chronic progressive external ophthalmoplegia. So this is primarily a condition that affects the muscles of the eyes, but can also affect the limb muscles as well. So they took the DNA from a muscle biopsy from those patients, and then they did a southern blot. So they linearized that circular genome using different enzymes, so to linearize it, and then they ran it on a gel, and this is a southern blot. And what you can see here are different populations of mitochondrial DNA. So when you run the DNA on this gel, it's sort of the smaller the length of DNA, the faster it runs. So you can see these bands here are one population of mitochondrial DNA, and this is another, and this one's a longer mitochondrial DNA once it's linearized, and so it doesn't run quite as far. So you should only have one population. They should all be the same length. And so you can see you've got controls here that have just the one mitochondrial DNA population, and these are patients that have these two populations. And the second population has run further because it's shorter when you linearize it. And the reason it's shorter is because there's a very large piece missing. There's a deletion of that mitochondrial genome. So, and that deletion often spans thousands of base pairs. And so you've got a much shorter DNA molecule that runs further on the gel. And what's interesting here is some of these patients, you can see they did both blood and muscle. And in the blood, I think over here, you can see they've only got one population, but in the muscle tissue of that same patient, that's where you can see these two populations. And this really exemplifies one of the characteristics of mitochondrial DNA in blood, in that because blood is rapidly dividing, there's this negative selection pressure against these deleted mitochondrial DNA molecules, and actually probably over the age of 5, you don't really see them anymore in the blood. But in muscle tissue, which isn't dividing, it's post-mitotic, they're retained, and you can see this deleted mitochondrial DNA population. And this is why, as many of you will be familiar, often we have to look in muscle to exclude these large-scale rearrangements in particular. And the other nice thing that this paper shows, so this was in 1988 in Nature, is that some of these, you can see in some of these patients, the deletions are running at slightly different levels. So that's because the deletion are different sizes in different patients. So some patients have a bigger deletion, and so a shorter mitochondrial DNA linearized fragment which runs further. And some of them seem denser than others. And again, that's really showing this concept of heteroplasmy, which I'll talk on in a second, but that's suggesting that population of deleted molecules have a higher percentage than others. So, for instance, this one, there's obviously a higher proportion of the mutated DNA than this patient here. And so since this discovery of these big deletions, there have been hundreds, literally, of pathogenic variants identified in this small circular genome. So these are all SNVs, so these are point mutations in the mitochondrial genome, which are maternally inherited, as many of you know, unlike these big deletions, which are often sporadic. And they're all associated with different clinical phenotypes. Some mutations are associated with different clinical phenotypes. Some phenotypes are caused by different mutations. So very, very heterogeneous, both clinically and genetically. And the reason that such a small genome has so many mutations, well, there are several reasons. It's got a mutation rate which is probably 20 times the nuclear genome, and that's because it has quite poor repair mechanisms. So when it develops mistakes, it can't repair them. It develops quite a lot of mutations because it's sitting very close to, it's anchored to on the inner mitochondrial membrane, close to all of those enzymes involved in oxidative phosphorylation. They're producing lots of reactive oxygen species that can damage DNA, and then it can't repair itself. And it's not surrounded by protective histones, as with nuclear DNA. It has a sort of rudimentary structure called a nucleoid, but it doesn't protect it quite as well, so it's got a very high mutational rate. So I've already touched on maternal inheritance. So your mitochondria all come from your mother, and so if your mother has a pathogenic variant in her mitochondrial DNA, in particular a point mutation, it will be passed to all her children. What is more difficult to predict is how much is passed to children, because, as mentioned, each cell contains hundreds to thousands of mitochondria, and therefore you can end up in a situation where you have a mixed population of mitochondria, some of which, in this case, the red mitochondria, have the pathogenic variant, and some, the blue ones here, that don't, that are wild type. And you can get this mixed population of mitochondria in different tissues. So some tissue is going to have a higher level of the mutant mitochondria than others. And what you end up with is a sort of biochemical threshold whereby at a low level you don't have any problems, but the more mutant mitochondria you end up with, the more likely the cell is not to work properly because there's not enough energy. And then you hit this critical threshold where the cell fails. And that threshold can differ according to the mutation that you have, one of those over 250 different mutations. Some of them have a different threshold than others, and also the tissues. So some tissues seem to be more susceptible to a reduction in ATP, and so this threshold might be further to the left-hand side. So that's heteroplasmy, this mixed population of mitochondria, and the threshold effect. And then finally, there's this bottleneck, which goes some way to explain why you can see such variability with mitochondrial DNA-related mitochondrial diseases within the same family. And what happens here is that at the level of a primordial germ cell here during embryonic development, there's a significant reduction in the number of mitochondria during development of the primary oocytes. And this is a sort of random segregation that occurs at the primary germ cell. So you can see, this egg here has quite a high proportion of a low proportion, of the mutant mitochondria. The mutant mitochondria, this has a higher proportion, and then there's an expansion as the eggs mature, and you can end up with this ovary that contains a population of eggs with very different levels of mitochondrial DNA mutation, and depending on which of those is fertilized, you can end up with a much more severe or less severe clinical phenotype in the child. And so even within the same family, you can get children with variable clinical phenotypes from no real problems to quite severe problems. So that all relates to mitochondrial DNA, but there are many, many genes within the nuclear genome, and we think probably up to 1,500 nuclear encoded genes that produce proteins that are important for the mitochondria to function properly. And many of those genes are relevant to maintaining this circular DNA molecule in terms of its replication, because it's constantly dividing and replicating, and transcription and translation of the mitochondrial proteins. And as I've already mentioned, that encode subunits of oxidative phosphorylation, and obviously mutations in any of those nuclear encoded genes can also cause primary mitochondrial disease. But obviously, rather than being maternally inherited, they follow Mendelian inheritance patterns. And of those 1,500 genes, we've identified more than 400 that are associated with primary mitochondrial diseases. And what we tend to see is children are more likely to have a mutation in one of those nuclear genes, whereas adults presenting later on tend to have a mutation in the mitochondrial genome. And what's very typical, as you'll all be aware, for primary mitochondrial diseases is they can cause all sorts of problems, because obviously all cells in the body have mitochondria, so they're not producing energy. In theory, any cell can be affected, and this is just a list of some of the clinical manifestations of primary mitochondrial diseases. But perhaps what's less easy to understand is why often what we see is tissue-specific manifestations of mitochondrial diseases, particularly when you know that mutation is across all tissues, but for some reason it's only manifesting in one or two, and that's something we tend to see more in adults. And so these are some of the common clinical syndromes that have been described historically, and you can see many of these as sort of acronyms. And to be honest, we're moving away from these syndromic diagnoses as we understand this group of conditions more and more. As we built up large cohorts, which has been very much helped, as Mike's alluded to, through the three different services, we've built up large cohorts of patients, and we get a better understanding of how these conditions affect people. We know that the sorts of problems that people have are not necessarily limited to these descriptive terms, for example, myoclonic epilepsy and ragged red fibers. We know that patients with that condition have a more extended phenotype outside of that. And so we're seeing much more overlap between these different conditions, and some patients don't fulfill the criteria, and so we're moving away from those and much more towards a genomic first description of the different problems that people have. And we're seeing many more phenocopies of these conditions, so mimics of primary mitochondrial diseases that aren't caused by a gene relevant to mitochondria. That might be a separate gene that's not relevant to mitochondrial function, but presents very similar, or even double dual diagnoses that mimic mitochondrial diseases. So we're sort of moving away from these descriptions. But certainly what we do see is this sort of change in terms of age of onset between much more severe disease that presents earlier in life and tends to be more multisystem. And as you get older, the older you present, you tend to have, obviously by definition, a less severe disease that tends to be more tissue specific. And certainly in adult patients, the most common manifestation we tend to see is neuromuscular, although we also see other parts of the nervous system implicated in primary mitochondrial disease. And the most life-threatening, arguably, in adult mitochondrial diseases tend to be stroke-like episodes. And as mentioned, there does tend to be this divergence in terms of the culprit genome, depending on whether you have an early onset or a late onset disorder, with nuclear genes tending to cause pediatric mitochondrial disease and mitochondrial DNA-related syndromes being associated with adult disease. So in adults, one of the most common neuromuscular manifestations that we see in clinic is progressive external ophthalmoplegia and ptosis. So this is a slowly progressive muscle condition that affects the extraocular muscles and the eyelids, and so eventually people are unable to move their eyes and they develop drooping of the eyelids. And this is really the archetypal mitochondrial disease, in that it's incredibly heterogeneous. It can be caused by those rearrangements that I mentioned earlier, those single deletions, which tend to be sporadic, where you've got thousands of base pairs missing of the mitochondrial genome. It can also be caused by nuclear genes that we know are important for replication of the mitochondrial DNA. And what you see within the mitochondrial genome there is the accumulation of lots of different deletions, so multiple deletions, which accumulate during the lifetime of that individual. Every time the mitochondrial genome replicates, it develops a new mistake, a new deletion, and so you get this polyclonal population of deletions affecting the mitochondrial genome, but it's caused by a nuclear gene involved in mitochondrial DNA replication, and the most common one of those is the mitochondrial polymerase gamma, POLG. And it can also be caused by point mutations in mitochondrial DNA. So POLG is obviously follows Mendelian inheritance patterns, so autosomal dominant or recessive, for instance. But these point mutations in mitochondrial DNA, they're maternally inherited. So you've got sporadic events, you've got Mendelian inheritance, you've got maternal inheritance, all causing the same clinical syndrome, albeit some of these conditions can have manifestations beyond the eyes. So this really highlights the kind of molecular heterogeneity that we see in these patients. The other phenotype, just to pull out, is primary mitochondrial myopathy, and the reason for doing so is because it's relevant later on when we talk about biomarkers and trials. So many of the current therapeutic strategies for mitochondrial diseases and the clinical trials are focused on this group of patients. Partly because that's where the small molecule targets. There's muscle penetration and it improves mitochondrial function within the muscles. And so often the patients recruited to those clinical trials that you're going to hear about have mitochondrial myopathy. And that's essentially where the muscle involvement in the mitochondrial disease is the most prominent. And so they tend to have proximal muscle weakness, exercise intolerance and fatigue. But that definition is slightly difficult sometimes because lots of people can have exercise intolerance and fatigue, and it's quite a subjective clinical characteristic, and so sometimes that's affected the way that we recruit to some of those trials and maybe how well those patients are stratified. So that definition can be a challenge, but that's certainly a sort of important group of patients for clinical trials. So given that this group of conditions can present at any age and follow any course and affect any organ and have any mode of inheritance, how are we diagnosing these patients? Well, that's something you're going to hear about later on today. But certainly we've benefited hugely from having these NHS highly specialised services, which are NHS England funded, so they're centrally commissioned and they allow us to provide a clinical service and a diagnostic service, we run a service in London, and we have partners in Oxford and Newcastle providing a similar service, and we work very closely together. And so this is just shows you the team that provide the service here in London, the clinical and admin team, and that's comprising includes consultants in the clinic, both neurology and clinical genetics. We've got clinical fellows and nurse specialists, physios, allied health professionals, research nurses, and an admin team. But it's important to remember we also have a diagnostic team that cross-cuts biochemistry, so the neurometabolic unit and the neurogenetics unit to help support diagnosis. And again, you'll be hearing from members of these teams later on in terms of how we work together as part of the multidisciplinary team to achieve a diagnosis in patients as quickly as possible. So I'm going to stop there and hand over to Micol Falabella. Thank you very much.
So good morning, everybody. Today I'm going to present an overview of the experimental models for primary mitochondrial diseases. I'm going to present some limitation of these models and also which question each model can answer. So I'll just briefly introduce primary mitochondrial diseases, which you have just heard about. So these are group of heterogenic disorders, both can be caused by mutation in the mitochondrial DNA or in the nuclear DNA. and they converge and is shared by energetic defect. So we have a defect in the OXPHOS machinery with decrease of ATP production, which then affects mostly the mainly the high energy demanding organs, such as brain, heart, skeletal muscle, and liver. Now, these disorders are heterogeneous and are also particularly challenging to model. There are several layers that contribute to this challenge. The first is that obviously, they can be caused by either nuclear or mitochondrial DNA defects, and these two genomes, they follow different inheritance patterns. Then we have an additional layer of complexity, which is given by the mitochondrial DNA. So we just heard the mitochondrial DNA is present when it's mutated in this heteroplasmy level that can vary across tissue, but also across time. Another complexity is given by the fact that the tissue that are usually affected are the ones that are the hardest to access, so like brain or heart. We also know that mitochondrial diseases are quite dynamic, so they can change with development, they can change with age, and they can also change with metabolic stress. Finally, another layer of complexity is given by the clinical heterogeneity. So even the same variant can present quite differently, can have a different, affect multiple organs, but also have a different phenotype. So just to give an introduction on the complexities that to model this disease is that it doesn't exist a model that can represent the full complexity of these disorders. So each model can fit or can answer a specific question. Now, the question that we can answer are very diverse. So the more simple questions are about pathogenic variant, the pathogenicity of a variant. So in that case, we can use cellular models that can be patient-derived cells or iPSCs or engineered models. If we want to understand more the mechanism of the disease, again, we can take advantage of the cellular systems, also small organisms or mouse models. But then when it comes to the tissue specificity, then in this case we need more advanced systems that can be other iPSCs derived cells, organoids or mouse models, or also 3D systems. Again, when the question regards, for example, the development, the physiology of the disease, we need in vivo models. Also when it comes to therapy. So in this case, we can use different animal models that I will talk a little bit about. And then when it comes to translation, so to therapy development, we usually have a complementary approach, so using both in vitro and in vivo systems. Now, starting with the first system that we usually we try to reach for, this is patient-derived material. This is a summary of the material that we can get from patient, and it's quite useful to answer different questions. First, the one that usually we use the most is skin fibroblasts, in part because they are easy to access. They retain the patient genotype, and then we can also gain valuable information regarding the OXPHOS, but also regarding other aspects of the mitochondrial biology. One limitation the patient-derived fibroblasts is obviously that they derive from skin, so they don't represent the tissue target of the disease. The other limitation is that many times we see that these patient-derived fibroblasts don't show any phenotype, and this is can be variant-dependent or donor-dependent, or they have a very mild phenotype, or another case is that they lose the phenotype over time. So when we have these cells in culture, they might compensate, so they might adapt, upregulating alternative pathways, or they can lose the phenotype because they just undergo senescence. So these are a good starting model, but they're not usually the end point. Now, to give you an example of what we can, the information that we can gain with patient-derived fibroblasts, I'm just going to present a work that was published, that we published last week. This is a work in which we were studying a variant in a complex 4 subunit, COXFA4. We had 13 individuals, 12 families, and the phenotype was mainly Lee-like syndrome phenotype. Now we had 6 lines available, 6 patient-derived fibroblasts available, and what we were able to do with these lines was to understand the disease mechanism. What we observed is that this variant was leading to a loss of COXFA4 protein and an upregulation of COXFA4L2, which is a paralog of COXFA4. So these two proteins share a high percentage of identity and similarity. And we also observed that there was a decrease in complex 4 activity, although there was around 40% of complex 4 activity retained. So these data, together with other data that we collected on patient-derived fibroblasts, showed that COXFA4 suggested that COXFA4L2 is responsible to preserve the residual complex 4 activity. Now, this model was very useful to understand the disease mechanism and to reveal a novel compensatory response. But again, the phenotype was a Lee-like syndrome, so we wanted to use a model that was more neuronal-like. So what we did was a very simple model. This is one of the simplest models that you can do. So we use an SY5Y cell line, which is a neuroblastoma cells. We knock down COXFA4. And as you can see here, I mean, when you knock down, so you have less COXFA4, and then you have an overexpression of COXFA4L2. So this was a nice model that showed that this what we observed in the fibroblasts was also recapitulated in neuronal-like model. So again, this is just an example of how you can use different models to complement and to answer different questions within the same research. Now, this what I just presented was a nuclear variant, but when it comes to variants in the mitochondrial DNA, things are a little bit more complicated, so you have to use different approaches. Now, the simplest one that you can use in cells is, which is historically used, is these hybrids models. So these are usually cells where you have a recipient cell that historically is the 143B osteosarcoma cell. You deplete the cells of mitochondrial DNA, and you can then fuse the cells either with platelets, which are like naturally like enucleated, or with a cytoplast donor. In this case, then you obtain what it is a hybrid, so you obtain a cell line that has a stable nuclear background, but then you can study the effect of the mitochondrial DNA variant. So this is a quite useful model, but at the same time, I mean, the limitation is that, again, the nuclear background is artificial, and then, I mean, it's not tissue relevant. So talking about tissue relevance, when you want to study, we know that primary mitochondrial diseases can affect different organs. So to study the relevance, the tissue relevance, I mean, you can develop different kind of models. Particularly, you can use iPSC-derived cells, you can use organoids, you can use the 3D systems. These are high relevant, but at the same time they have a limitation. In part is that they are immature, so what you get is not, they don't mimic the adult tissue, but they usually mimic an early embryonic or early fetal stage of the tissue. They're also quite variable, and in terms of cost and time, they require they're quite expensive and also time-consuming. So again, these models are useful. They are more relevant to the human tissue, but they also require the very careful validation. Now, moving from the human cells and the in vitro model to in vivo model, we have several models. We have more simple models, which are like yeast or C. elegans, which I know that for the clinical audience, they kind of quite far from from clinics, but at the same time, I mean, they are quite useful when you want to answer very simple questions. So if the mechanism, the mitochondrial mechanism that you want to study is quite conserved, these two models are very useful. Are also useful for genetic screens. At the same time, I mean, the limitation is quite obvious, so they are like, they lack the clinical and the tissue relevance. Then we have models that can answer developmental biology questions like C. elegans. We have Drosophila, which is quite useful when you want to study neuromuscular phenotypes. Zebrafish, which is a quite useful model both to test therapies, but also for developmental biology question. And then finally, we have mouse models, which are mammalians model, and they can you can use these to track disease progression, but also to test preclinical therapies. So as you go from more physiologically relevant models, what you can see in this graph is that you lose the speed, you lose also the scalability and the simplicity. So again, you have to choose, even when it comes to in vivo models, is like the most important thing is to have the right question in mind and then to use the right model to answer that specific question. To give you another example about how we can use these models and combine these models, both patient-derived models and in vivo models, I'm going to present you another study that we published last year. In this case, we had a variant in PTPMT1. So PTPMT1 is a gene that is involved in the cardiolipin biosynthesis. Cardiolipin is a specific phospholipid that is present only in the mitochondria. And in this case, we had 6 individuals, 3 families, and the phenotype was a neurodevelopmental phenotype. We had patient-derived material, and we used this patient-derived material to understand the mechanism of this disease. So we used patient-derived fibroblasts to basically check the presence of the protein, and as you can see here, I mean, this top band, this bottom band is absent in the patient fibroblast. We then assess the cardiolipin abundance in skeletal muscle and dry blood spot, and as you can see, I mean, there is a significant decrease in cardiolipin. We also use this model to characterize the morphology of the mitochondria. And this is, so you can see here that the mitochondria appear more fragmented, and we also use this readout to rescue the patient fibroblast and then confirm the pathogenicity of the variants. We also observed that there was a decrease in complex 1 activity, and this was measured in skeletal muscle. So patient-derived material was quite useful to understand the mechanism of the disease, but at the same time, as I mentioned, the phenotype was neurodevelopmental, so we wanted to see what's going on in vivo. So to do so, we developed a zebrafish PTPMT1 knockout model. As you can see here, I mean, it's quite clear that at 19 days post-fertilization, you can clearly see that this zebrafish is much smaller, and also it lacks the anterior chamber, which is an indication that there is a developmental abnormality. So we then perform additional biochemical tests on these models that match what we found in the patient fibroblasts. So this is an example, I mean, where you can combine different models to answer like a bigger question. So patient-derived for the mechanism, and then in vivo model to understand the consequences. Now, I talked about the model, how you can use, how you can complement and use different models. But one important thing to keep in mind is that each model has its own limitation. So first of all, there is the tissue mismatch. So I talked about fibroblasts, but obviously what you can see in fibroblasts, it cannot resemble what happened in the target tissue. There are also culture artifacts, and this is something to keep in mind because mitochondrial disease are metabolic disease. So when you have these cells in culture, I mean, depending on the media that you use, on the components that you use, you can change the bioenergetics. Obviously, I mean, this is quite obvious, there are species differences, so sometimes it can happen that even mouse models, they don't resemble the clinical phenotype. And then we have misleading readouts. So this is particularly important when it comes to therapy. So when you have also a biochemical improvement, this doesn't necessarily mean that there is a clinical benefit. So again, every model is informative, but it has its own limitations. And this, is particularly important to have in mind when you start when you have your clinical and biological question. So just to finalize, the take-home message that I want to give with this talk are just four. So first of all, is that when you have nuclear DNA or mitochondrial DNA, they require different modeling strategies because they have different biologies. Patient-derived cells are usually the first starting point, but rarely are the end point. So you have to complement with additional models. And then, there is no best model for primary mitochondrial disease. The best strategy is to try to find, to combine complementary models. Now, one last take-home message is that usually the best model is not the most complex one, but is the model that can answer best to your question. And this is actually the end of my talk, and thank you so much.
Okay, thank you very much. I'm not going to repeat once again the features of mitochondrial diseases because the first two presentations already gave you all the information. I want just to flag a couple of points that will be needed to follow my presentation later on. And the first point concerns the complexity of the clinical syndromes in mitochondrial diseases, and they can have many different manifestations and different organs and systems can be affected. And the second point concerns the genetic complexity of these conditions. Over 400 genes, as Rob already mentioned, have been found as responsible of mitochondrial diseases, and this table summarizes, which is taken from the Genomit Consortium. You can freely look at it online, but over 400 genes have been described. So, as you can easily understand, this poses incredible challenges in terms of developing new therapies for mitochondrial diseases. Currently, there is no really curative treatment for these conditions, and that's what we have been doing over the past 20 years in my lab. Given this complexity, we can identify two approaches to tackle the problem of developing new therapies for mitochondrial diseases. And the first approach is a very general approach trying to treat more than one single condition, and the other one is a disease-specific approach. There are several ways in which we can take to modify the course of the disease. And some of these are listed here. They include the stimulation of mitochondrial biogenesis, the activation of mitodynamics or autophagy or apoptosis, and we'll get back to this point in a second. And the other way is to correct mitochondrial DNA mutation post-transcriptionally. And obviously gene therapy is a possibility to correct disease or mutation-specific conditions, and we'll get back to this point. So this is a table that summarizes the same concepts, and basically what I would like to focus on in the rest of my presentation is a couple of examples taken from my own activity, research activity, focusing on the stimulation of autophagy or mitophagy as a way of improving mitochondrial diseases, and the other one is based on AAV-based gene therapy. But actually several approaches have been proposed by us and several other laboratories, including nucleotide supplementation, stimulation of mitochondrial biogenesis and mitochondrial shaping, bypassing OXPHOS defects, shifting heteroplasmy, and you'll probably hear something more in the next presentation, and the elimination of toxic compounds. As you can see, understanding the basis, the pathogenesis of mitochondrial diseases has been fundamental to try to develop new therapies. And we made good progresses from this point of view, but we still lack life-changing therapies. So the first example I wanted to give you is based on the stimulation of mitophagy, which is a mitochondrial quality control pathway, according to which damaged mitochondria are progressively engulfed in autophagosomes and addressed to degradation by fusing with lysosomes. So the idea here is to get rid of dysfunctional mitochondria within cells and tissues. One way of stimulating mitophagy is to use a compound called urolithin A. Urolithin A has been known for quite several years to improve mitochondrial health, whatever this means, but in principle this is obtained by stimulating mitophagy. Urolithin A is a natural compound which is particularly enriched in red fruits and nuts. Actually, this food contains the precursors, which are called L-gitanin and ellagic acid, which is then converted in the gut by the commensal bacteria in urolithin A, which is then absorbed and goes to the tissues. We studied the effects of urolithin A in a mouse model we developed many years ago, which lacks an assembly factor for cytochrome C oxidase, which is complex 4 of the respiratory chain, and this gene is called COX-15. Actually, it's not really an assembly factor, but it's an enzyme which is involved in the biosynthesis of the prosthetic group of cytochrome C oxidase, heme A. So basically, if you look at this, it converts heme O produced by COX-10 into heme A. And the mouse model develops a very severe myopathy characterized by exercise intolerance. This is how much a knockout mouse for COX-15 can run on a treadmill compared to the wild type. So you see that it's very, motor activity is very reduced. Complex 4 is very low compared to a wild type in skeletal muscle. Just to be more precise, this is a muscle-specific knockout. So in other tissues, complex 4 is normal. And what is absolutely evident in electron microscopy is the accumulation of these gigantic and deeply altered mitochondria. And we gave urolithin A by gavage for 10 weeks to these animals. And what we observed is that after about 5 weeks, the treated mice, they started to run a little bit more, although this difference is not really significant. But after 9-10 weeks, 9-10 weeks, they started to run definitely more than untreated or vehicle-treated controls. And if we look at the histology of the skeletal muscle at the end of this treatment, you see that there is an amelioration of the myopathic phenotype with an increase in the cross-sectional area, although we don't see much of an effect in the complex 4 activity. But what became evident by looking at the electron microscope is that the gigantic mitochondria that we observed since the first experiments on this model are not present anymore in the skeletal muscle of the treated animals. Although there are still many abnormal mitochondria, they look much smaller, and this is a sort of quantification, and also we saw the appearance of definitely normal mitochondria. This is the wild type. This is urolithin-treated animals. So there is clearly a clearance of dysfunctional mitochondria. So to try to explain this, we studied mitophagy, and the way we did this is to cross the COX-15 mice with a reporter mouse for mitophagy, which is called the MitoQC mouse. And the MitoQC expresses a mitochondrial targeted GFP, RFP protein which is targeted to the outer mitochondrial membrane, and when mitochondria are engulfed in the autophagosomes and fused with lysosomes, the acidic pH quenches very quickly the green fluorescence of GFP, and the mitochondria appear as red dots. You can't probably see much from the fluorescent image here, but we definitely saw an increase in the number of red dots in treated skeletal muscles. And this is the quantification again. Red dots are less in the knockout mice, and they are increased to normal levels in the knockout mice, suggesting that actually there is an activation of mitophagy, and this is possibly a cause of the clearance of dysfunctional mitochondria. So I'm now moving to the second part of my talk about AAV-based gene therapy. AAV stands for adeno-associated viruses. These are small viruses which are replication defective, and they were actually isolated as contaminants of adenoviruses, so because they require the machinery of adenoviruses or other viruses for replication. They are internalized in cells upon interaction with the surface receptor, and then they go through a clathrin-mediated process, and they undergo the endosomal pathway, and this step is essential for the activation of the virus, because the change of pH in the endosomes activates the virus, which then can either be degraded through the proteasome pathway or free the genomic content, the DNA, in the nucleus. Natural AAVs contain single-stranded DNA, which needs to be copied into the second strand to get a functional genome, and this step is rate limiting for the replication of the virus or the expression of the transgene that replaces endogenous genes. However, in the lab, this genome has also been engineered to be double-stranded, and this is called self-complementary because it forms this sort of hairpins which facilitates the expression of the transgene. A number of natural variants exist of AAVs, which are called serotypes, and these serotypes can preferentially target different organs. In case of rodents, for instance, AAV8 preferentially targets the liver. AAV9 has a much more widespread preference, so it can target different tissues. Obviously, these are viruses, so they trigger an immune response, but they have several advantages. In particular, they can survive for a very long time in non-replicating tissues. They are relatively low immunogenic compared to other viruses, although they're still viruses, as I already said, meaning that they can be used only once, basically. They are not pathogenic in humans, and they remain largely episomal in our cells. That means that they do not integrate in the cellular genome. So we started using AAV viruses quite a long time ago, and we initially focused on a mouse model of Lee disease, which lacks a small subunit of complex 1. This is respiratory complex 1. This subunit is called NUFS4, which is located right here, this purple subunit. The mouse is a relatively good model of the human disease. It develops a rapidly progressive encephalopathy, you can clearly here distinguish the knockout mouse. Here it's still in a relatively good shape, but the mutant mice do not survive beyond 7 weeks of age. So after many trials and errors over the years, we came out with this strategy which gave the best results in terms of improving the phenotype by injecting self-complementary AAV9 re-expressing the human gene in the in the cerebral ventriculi, and systemically in newborn mice. And this led to a robust increase in survival of these animals. So untreated mice die by 7 weeks of age, and treated animals can survive at least 6 months. We then terminated the experiment. They largely prevent neurodegeneration. I'm just flagging this pathogene staining, which is specific for dying neurons, which are not present in treated animals. Complex 1 activity is largely restored, almost completely restored, by spectrophotometric assays in gel activity, and this is a corresponding blot. In the knockout, you see the accumulation of this lower band, which is almost fully rescued. It almost disappears in AAV-treated animals. So as the brain is a preferential target for mitochondrial dysfunction, but it's not the only one, and targeting the brain with AAV is still a huge challenge. So we more recently moved to different serotypes to treat muscle disease. And this is a COX-15 knockout mouse treated with the myotropic AAV, so AAV which is specific for the skeletal muscle. And you see that treated animals started to run much more than untreated controls. After about 4 or 5 weeks, there is a partial recovery of COX activity, and here you can appreciate the distribution of the virus. And finally, I wanted to flag this information because it's important that the amount of virus we used in our experiment is by far lower than the one used by some colleagues in the States, using the same serotype to treat a different mouse model, but that's the advantage of using muscle-specific, liver-targeted serotypes. I'm just very briefly mentioning that more recently, in collaboration with Lucas Corrano and Martina Semenzato in Padova, we developed a heart-specific knockout for COX-15 by using an inducible, heart-specific and inducible recombinase, which leads to the development of dilative cardiomyopathy. You can clearly see that the heart of these animals is enlarged, and this is the quantification. And we treated them with AAVs, again, to re-express the missing gene, and you can appreciate that now the size of the virus is almost normal, and this is the quantification. So this is my last slide. I just want to thank my group and my collaborators and you for the attention.
Good morning. Pleasure to be here, especially on Tuesday after bank holiday weekend. So what I'm going to do in the next 15 minutes or so, I'm going to tell you a little bit more of the advances we're making in the engineering of the mitochondrial genome, both in my lab and so thanks to colleagues in the field. So just a reminder about the mitochondrial DNA being multi-copy. That presents a little bit of a challenge in terms of engineering. We have rather efficient copy number control. So if we regulate, if we have a mixture of 2 genotypes, if we can sort of deplete one of them, the other will pop up again. So that's quite important for the concept of this talk. And obviously we have homoplasmy, all genomes the same, heteroplasmy, mixture of 2 genomes within one cell. And this biochemical threshold, some levels of empty DNA mutations are tolerated in terms of pathology. So then I'd like to give you a little bit of the overview, at least how I see it, why engineering of the mitochondrial gene has been challenging over the years. I think one of the key reasons is that we are unable to import nucleic acid into mitochondria. So we cannot essentially replace this large plasmid within the mitochondrion because we cannot reach the matrix in the mitochondria. Also, many of the genome engineering technologies, they require a donor copy next to the recipient copy of DNA. Again, even smaller fragments are not possible to be delivered into mitochondria. So for the same reason, not being able to import nucleic acids in mitochondria, CRISPR-Cas9 revolution has not been sort of adopted to work in mitochondria. So we cannot deliver RNA, therefore CRISPR-Cas9 needs guide RNAs to work, and therefore we were unable to use all these advances CRISPR-Cas brought to life about, 13 years ago or so. So as a field, we've been creative, and what we've been doing is trying to randomly mutagenize the genome and then select useful genotypes and phenotypes. And these methods have been very successful. However, the limitation there is that we don't have any control into a site we are mutating. So sort of on the contrary, we were quite successful in deciphering methods which allow us to regulate the numbers of genotypes of particular mtDNA. So essentially to regulate heteroplasmy. And we've been achieving that by creating cut into a specific genome and therefore changing heteroplasmy. And we can do it both ways. We could either cut mutant mtDNA to reduce pathogenic heteroplasmy. We can also cut wild-type genomes to increase mutant load, and I will show you some examples later on. And then, very excitingly, about 5 years ago, there's been emerging tools which allows us now to change specific bases in the mitochondrial genome, so-called base editors. So I'll try to give you a little bit of flavor of each of these strategies and how they help us developing models of mitochondrial DNA dysfunction. So first I'd like to mention these phenotype first models because they've been quite useful in the field. There are a couple of examples coming mainly from the work of Niels Golar Larsson and Jim Stewart when they were both in Cologne. Now Jim is in Newcastle. So what they've been doing, they develop a mouse which has a proofreading defect in the replicase in the mitochondria. So there is a polymerase that makes many mistakes, and it's called mutator mouse. And then by clever back-crossing of these animals, they were able to derive animals that contain only a limited number of mutations, preferably one mutation. And then the key examples there are mutants in tRNA alanine, which is encoded in the mitochondrial genome, one in the position 5024, and when this mutation occurs, this tRNA is unstable, cannot sustain intramitochondrial translation, and these animals develop some mild cardiomyopathy and recently reported muscle problem. And a similar technology was used to derive a second mouse model heavily used in our field. It's 5019, this 5 nucleotides up in the tRNA molecule. And what happens in this model is that this tRNA cannot accept amino acid, cannot be charged readily. Again, translation in mitochondria is perturbed, and then these animals have a slightly more severe phenotype than 5024. And I'd like to mention that generation of these animals has only taken 15 years because of the generating the mutagenized animals, then backcrossing, and then so sort of as a field, we've been working very hard to try to develop methods that would allow us to make these models a little bit more faster. So in order to engineer the mitochondrial genome, we've been repurposing essentially parents and grandparents of CRISPR-Cas9 technology. So what I'm presenting here, there are two architectures. The top one is the zinc finger based architecture, which allows us to target specific sites in DNA. And this specific site is sort of indicated by this gray sphere. So what we can do, we can now design these colorful blocks so that they bind different DNA triplets. And if we can array them together and sort of target both strands, we are able to target specifically the gap which is between these two monomers. And similarly, we can now design these tail blocks. The design is slightly different. Rather than targeting triplets of DNA, with targeting single base pairs, but essentially the architecture is the same. So what we can do, we can deliver DNA encoding these proteins into a cell. This DNA will also contain coding sequence for mitochondrial targeting sequence. Therefore, these proteins can go into mitochondria. And importing proteins into mitochondria is relatively easy. So what we can do using this scaffold, we can, for example, between these two monomers, we can introduce a nucleolytic domain. So the nucleolytic domain presented here is FOC1. It's a domain that comes from a restriction enzyme. And if we do that, what we can do, we can create double-strand break. We can essentially cut DNA in the sequence between the binding site of these two monomers. And this technology has been born about 15 years ago, let's say, and we've been able to cut different genotypes in the mitochondrial genome, changing heteroplasm. However, there's also meganucleases, which is essentially grandparents of CRISPR-Cas9, slightly more difficult engineering tool, but then still very potent in changing heteroplasm. So very excitingly, about 5 years ago, there is a new domain was discovered called DDDA, which comes from bacteria. And what this domain can do, it can deaminate a cytosine in DNA. So this chemical reaction here. And if we deaminate cytosine, we'll end up with uridine, essentially, which if a DNA replication comes through this uridine, this U will be read as a T. So essentially what we can do, by changing the chemical properties of cytosine, we could have C2T mutations. And this sort of opened up the field quite, and people got a little bit more inventive. And soon after discovery of these C2T editors, A2G editors were also described. The biochemistry behind is slightly different, but then essentially we again deaminate adenosine, which ends up with having inosine in DNA, which is read as a G. So we have A2G mutations. I won't be talking much about those, so I'm just flagging them that both C2T and A2G is possible. So just to break it a little bit apart, if we want to mutate the mitochondrial genome, all we have to do is to design these purple caterpillars around our favorite C, and then this C will become U, and after 2 rounds of replication, we'll have C2T mutation. And this tool is called DDCBE, from DDDA, so deaminase derived cytosine base editors. And I'll spend the rest, the next 5 minutes or so, giving a few examples how we can use this into developing, especially mouse model of mitochondrial DNA dysfunction. So one thing we've done in the lab was creating this MitoKO library. So what we've done, we've introduced premature stop codons by targeting one of the first tryptophan codons in the coding sequence of each of the 13 proteins in the mitochondrial genome, so that it becomes a stop codon. So essentially, we truncating proteins one by one. And we've done it for every protein in the mouse mitochondrial genome. So then we have a library that allows us to use plasmids in vitro to transfect cells to generate ablated proteins in mitochondria. Also in vivo use, so we can do embryo injections, somatic tissue-specific AAV delivery mentioned by Carlo, and working towards transgenic animals as well. And as a sort of spin-off, we have now a collection of cell lines with different heteroplasmy. So what we can have is a sort of the same nuclear background, which was already mentioned, that's a little bit of a challenge, with different heteroplasmy. So just an example of an animal models which we derive using this library. We introduced this premature stop codon in ATP6 gene, which is a subunit of ATP synthase or complex 5. Mutations in this gene has been reported in variety of primary mitochondrial diseases. So we've taken this base editor and deliver RNA encoding this base editor into one cell embryo, and then injected this, implanted these embryos into pseudo-pregnant female, and then have a founder animal with a relatively low heteroplasmy. But what we can do now is leveraging the bottleneck so that this animal can transmit mutation with different levels, and if we cross this animal with wild-type male, we can always take mom with the highest heteroplasmy. So we can selectively breed these animals to higher and higher heteroplasmies. So in F3, we had about 50% heteroplasmy, and now here I'm showing a result of a 4th generation when we're now looking into assembly of ATP synthase, and we do see that this assembly is perturbed quite similarly to a patient affected with mutations in the same gene, actually published here by Rob and colleagues. So, okay, another example. So then you could say, okay, ablating a gene is quite a severe way of causing dysfunction. How about modeling mitochondrial DNA mutations specifically? So here our focus switched towards Leber's hereditary optic neuropathy, LHON, which is a disease affecting complex 1 and due to problems with retinal ganglion cells and optic nerve atrophy. And so there are main mutations causing this gene, with these 1177As being most frequent and more severe with bad prognosis. And at the moment, there is no mouse models for this disease, also lack of curative treatments. And there are still several key outstanding questions. Why about molecular mechanism for this disease, the nature of cells being affected, why there is a sudden death for these cells, and so on. And so we thought that a mouse model could help us tackling these questions. So we designed these DDCBEs, cytidine base editors, tested them in cells, and what you can appreciate here, this is a mouse numbering, so essentially this is very well conserved, this site here, and after single delivery of base editors, we can create about 60% heteroplasmy, whereas, with these base editors being quite precise. All these black dots here, they show us a low level of other mutations being introduced. So here again, we introduce this base editor into mouse embryo, the very similar strategy as for ATP6 mutation, and we could have animals with F2 with about 30% of LHON mutation. So we've been selectively breeding these animals. Now we have animals with about 70% heteroplasmy, but as I told you, mostly these mutations are homoplasmic. So then how can we make this mutant homoplasmic, 100% of mutation? So then the new technology of introducing relatively low levels of mutation, combining with the older technology of being able to selectively shift the heteroplasmy, could be combined. So essentially the strategy is as follows. We introduce a low level of mutation, and then we want to cut out wild-type genomes using these specific nucleases. So again, this is the same cell line I showed you before, but now this cell line is treated 4 times with a nuclease that eliminates a wild-type molecule. So at least in cells, we can achieve homoplasmy within essentially weeks. So now what we're working on, and it's sort of becoming our pipeline, is that we do have our animals with relatively low levels of heteroplasmy, but then from one of these animals, from mom, we take embryos, in this example 30% of heteroplasmy, and now we're treating this embryo with a specific nuclease that can reduce wild-type DNA. And then this should help us, expensive and time and lengthy selective breeding, and we could reach the high level of heteroplasmy animals soon. So we are not the only ones. Here I'm listing probably not complete list of animal models which have been developed using base editors. So they all contain mutations which are relevant for pathology in mtDNA. And I'd like to flag out, and I think Sara will tell us a little bit more about it, that some of our models are available within the National Mouse Genetic Network and MitoCluster. So as a summary, I'm very excited to see how the field is developing and how easy it is to now generate mouse models. Obviously, this can be translated into organoid models as well, and the other chassis. And then I think on the future outlook, what we need to work on is to make the A to G editors a little bit more precise, because they are still, they do not have single nucleotide specificity. And I think having animals that can introduce, where mutations can be induced into tissue-specific manner, will be very useful. And then Krelog systems of conditional expression would be very useful. And I think working on the models of large-scale mtDNA deletions, Rob also mentioned, would be very useful, as at the moment we don't have those neither. So with that, I would like to thank my team, especially Lucia, who is driving the LHON yet unpublished work, and others as well, and also all my collaborators and many institutions for paying our bills. And thank you for your attention. Thank you.
So we've heard lots about modelling and about different experimental models. So I'm going to focus on the use of animal models, specifically mouse models in research, and just some ideas for the future as well, because I think the mouse has been around as an experimental model for many years, but we're really on the verge of having technologies which could really revolutionise how we use the mouse and how we use other model organisms, both in terms of making the best use and gathering the most amount of data from our mouse models, but also making them really relevant for human disease. My home team is the Mary Lyon Centre, which is the hub of the National Mouse Genetics Network. And what we really do as a whole living is that we try to enable mouse genetics research. And so we spend a lot of time with people who you've heard this morning, who use mouse as one of their everyday models. But increasingly, we're spending lots of time with people who have never used an in vivo model, but have got their experiments and their work to a stage where there is no other model which is going to answer their experimental questions. So there's no cellular model which completely captivates everything that they need to answer, and they need a whole cell physiology model. And I think that this is a really important thing to emphasise to everybody. No research starts with an animal. No research on animals happens unless we can find if there is an alternative for using, if there's cellular models, if there's the kind of advancing organ on the chip models. But there is still a humongous need for these in vivo models, and we'll show you how we're trying to develop these. We partner with all sorts of organisations as well, academic and commercial organisations, and kind of a place where you can come together of different entities to try and answer the same question and answer pertinent clinical questions. And one of the things that we've been doing, and this kind of gains an ever more acceleration in our thoughts, is making sure that our models are really relevant to human disease. We do some discovery research as well, but really the Maryline Centre focus and the focus of the field has really moved towards the relevant to human disease. And that's been because there's been lots of commentaries around, and lots of you would have read commentaries around the relevance of models and the lack of translatability of mouse models into human patients when you're talking about therapeutic testing. And we really have been working on some of the criticisms of these, and they're very good criticisms from where the field were, and they basically highlight, this is a paper that basically highlights two things, that says that sometimes the diseases in the mice aren't caused by the same thing as they're caused in the humans, and importantly, sometimes the way that we diagnose this in a mouse, or we look at it in a mouse, isn't the same way as you look at it in the humans. So you can't map biomarkers that you might see in a patient group very well over to experimental models, and these are gaps that we really need to reduce when we're trying to drive to increase preclinical validation. And so this term, kind of disease positioning, has been used a lot in the field recently, and it's all about trying to develop mouse models that replicate specific human diseases. And this is by selecting or generating the right mouse models genetically, by characterising them in the right way, and having validation platforms which cross-compare human and model organism data, and therefore like aligning the features of and in this case I'm going to use a mouse model because I'm a mouser, but really any animal model with the scientific and therapeutic goals of the research. So this really starts with us, with generating the right model. And genome engineering, you've seen some really clever genome engineering in the mitochondrial. I'm going to take you through a little bit of what we can do with the nuclear genome. And CRISPR-Cas9 really blew our minds and revolutionised the field. Was 13 years ago or so that it first came onto the field, but it's, obviously takes some time, and it takes some time to get into a scale where you can be really efficient. And if you'd have taken us back 15 years or even sort of 20 years, it would have taken, 2 to 3 years really to have a colony of knockout mice that were available for people to do experiments on. And now we can sort of generate null animals, so we can knock a hole in the genome anywhere and have mice ready for you to use well within a year. The same for point mutations, so mutations from patient groups, and quite often this is some of the work we do, is we have mutations from patient groups that need to be recapitulated if there's the relevant sequence in the mouse. Not always the relevant sequence, not always the relevant gene, but when there is, this is some of the work we do. This can also be delivered within a year to have experimental work. But we can also use CRISPR-Cas9 for some really clever things that tell us not only about the disease phenotype, but also start to tell us about the mechanism, start to tell us about the cellular physiology as well. And we can attach all sorts of tags to genes, and we can attach all sort of genetic tools that have been described in the talks beforehand, recombinases, markers, reporters. And we can also insert or delete any cell type that you want, because we can rapidly engineer animals that have all of these genetic tools. So now we can rapidly, within a year, like a year is probably do it within 9 months if the experiment's easy and the target's easy, deliver mice where you can have pieces of your genome missing. We can add additional pieces of genome. We can have point mutations. In a little bit longer, we can develop complex genetic tools like Carlo's been explaining you, where we can take the gene in and out at different times in different tissue types. On the horizon for us, and these are some tools that we're starting to use, are kind of other tools other than the classic genome engineering, and that are the tools of the integrases. So basically, you engineer a mouse strain to have a landing pad, and then you can insert very large pieces of DNA into those landing pads. There's a technology called Chrysomere, which means that we can use CRISPR to introduce quite big chromosomal deletions. So we're not just talking about kind of single nucleotides now, we're talking about big chunks of the genome that we can invert and we can duplicate and we can delete. And then an extension of that is where you want the gene to perform normally, but you want to reduce that the protein that the gene is producing. It has to be a protein coding, obviously, but the protein that the gene is producing, you want to reduce that at specific times. And there are technologies called Degron technologies, which are being used in vivo now, and which are being developed to enable you to give a ligand to an animal and reduce the protein, but when you remove the ligand, the protein rises again. So it really would give us some very good insights into the mechanisms. And just to show how this can sort of align with human diseases, this actually is a mouse disease as far as I'm concerned. So this is a point mutation in cadherin 23 that most of our mouse strains contain. And above the panel on the top is wild type that shows that the hair cells in the inner ear have been lost when they get older. So this is kind of 6 months plus, so the mice are deaf. But if you replace that single nucleotide with the wild type allele, so that with the functional protein, you restore hearing in these animals. We also have lots and lots of different models where we use combinatorial approaches, and that means that we might have one gene that's knocked out, one gene that's overexpressing, one gene that the protein of which we're moving up and down depending on the timing we need to, and one we're taking out in a different tissue. So now we can bring together all of these different genes that act together to produce a gene. We can try to recapitulate that in an animal. We can also, this is slightly controversial, but we can also do some work around genetic diversity, not to be confused with human genetic diversity, but we can introduce different mutations on different background strains that are genetically diverse, and that will tell you whether that mutation is modulated by things in the genetic background of these strains, and therefore is susceptible to genetic modification. It won't tell you anything about the diversity of the human genome, but it will introduce that element of, can this gene be modulated by other genes in a background strain? One of the things that thanking Michal just told us about was the fact that the Mary Lyons Centre has a national archive for mouse strains, and we have literally thousands of mouse strains that we send all around the country and indeed all around the world in quite an efficient service. So if you do want a mouse strain, or you're thinking of a mouse strain, it's always worth dropping us a line to see whether there's something already generated that you can use for your studies, or one of your collaborators has already deposited something. But it's great making all of these strains that replicate the genetic disease. But then we have a mouse, and a mouse really isn't a human. They're nocturnal, they're prey animals, they act very differently. So we need to develop a much better way of characterising these animals. And over the last few years, we've been really accelerating the ways we're looking at sophisticated phenotyping tests. By the way, the best phenotyping test is somebody looking very carefully at your animals, whatever. But that's the best initial phenotyping test, is having very experienced people who really know what a mouse looks like and what a mouse behaves like. But we have also been developing, I don't know whether that will work, we've also been developing things using machine learning techniques, and using techniques where we attach landmarks to parts of the animal, and we can monitor how much these animals move. There's some huge advances in imaging, and especially in this kind of environment in London, there are some amazing imaging centres in London that can do very precise imaging of small rodents. And the thing in the middle is a recording of an ultrasonic. So there's lots of things about these animals that still need exploring. And just to say, the optical imaging systems are amazing for animals now, and are absolutely amazing. You can get very precise images of blood flow of neonate mice, and so they're really worth exploring further. And this is just an example of how the kind of AI machine learning technologies are being embraced by animal research community. And just to say, to get AI people interested in animal research is quite difficult. It's not the top of their list of what they want to do. But if you give them loads of data, they quite like it. So this is just some AI that we've developed with a company who were interested because they wanted to sell it, obviously, of a Huntington's model, and we've managed to measure how much the animals climb. And climbing on the roof of the cage is a passive phenotype, so we're not making the mice do anything. We're not putting them under undue stress. We just monitor them 24/7 for how much they climb on the roof of the cage. It's a voluntary activity, and it's also seen as a luxury activity. So I always say it's a bit the same as if you took me to the edge of the cliff and you went to push me off, I would hang on because my life depended on it. And that's what happens when you put an animal on a grid and you turn it upside down. It will hang on because its life depends on it. But if you put me in a play park and there were monkey bars, I'm too old and I'm too fat and I wouldn't bother. So it's kind of that sort of measure that we can tell whether the animals are strong enough to hang on for their dear life, but do they volunteer to do it? Do they want to do it? And it's all those kind of subtle intermediate phenotypes that lead up to you having a catastrophic disease. And we can see, the interesting thing is males and females are very different. So if you're doing any animal studies, it's really important to include both of those sexes. But we can see here from the arrow on the top set of the graphs points out the blue line is the wild types. And at the end of the night, the wild types are still climbing, but you can see on the orange line, the actual mutants decide not to climb. And this just kind of shows you how complex these phenotypes are. They're not, are you climbing, aren't you climbing? They're about a long period of time. At the beginning of night, they do all right. At the end of the night, they don't do very well. And so we need to use much more complex phenotyping tests to analyse these animals. So just to kind of finish off on what we talk about all of the time and is how similar are mice and animals? Well, cellular mechanisms are very similar. We have the same organs roughly, but the basal metabolic rate in the mouse is very much higher because of the surface area to volume ratios. It's about 7 times higher. Remember, mice are nocturnal, so if you look at them at 9 o'clock in the morning, they should be asleep, and they're probably going to react differently than they do at 9 at night. They are prey animals, so they're scared of us anyway, and however much you think you've trained your mice, they're still scared of you. And we keep them in very non-human conditions, so we keep them 7 to 7 on light, and we keep them on very sustained. They have ab-live food, and they have constant water, and are meant to be constantly not under any stress. So it's similar, the physiology is similar, but it's not quite the same. And what we've been thinking about recently is how do we start to change this, and how do we start to move, changing the norm from how we keep animals and to how they might reflect human disease better. And one of the examples is kind of exercise, and we're sort of working on how to do this. But we put a wheel in and we hope animals run on that, and we say that's an exercise challenge. But in actual fact, humans don't. An animal will run on a wheel all night. Humans won't exercise for 12 hours. Or on and off for 12 hours. They exercise in bouts, they don't exercise every day. They exercise to fatigue sometimes they don't exercise at all. So we need to sort of think about how we're going to start modelling that. There's been something talked about quite a lot in the community called thermoneutrality, and that's the kind of state that you can keep your mice in. So maybe a temperature you can keep your mice in 28, 29 degrees is kind of what is pitted as that temperature, where where they don't use lots and lots of their metabolic rate into keeping warm, and it makes their metabolic rate lower, and so it reflects human. So instead of being 7 times more than humans, the metabolic rate is reportedly to be sort of 1.5, 2 times humans. So it might sort of normalise that. That's a bit of a controversial field, and I'm not a metabolic scientist, so I'm not going to argue that out with anybody. But there are lots of people looking at the effects, and this is just a summary, looking at the effects of putting animals in warmer cabinets and keeping animals in warmer cabinets on different disease phenotypes and different disease models. And the one thing that we can say now is that there are significant reports that it really changes the disease phenotype in an animal. Oh, sorry, I pressed the wrong way. And then finally, the microbiome. I've been doing this all of my career, so I've been looking after animal houses all of my career, and about 25, 30 years ago, when all of the genomics was coming out, anything that couldn't be reproduced by different animal houses, everybody said, oh, it must be the genetic background of the mouse. And then we sequenced the mouse, and then we realised we could keep that constant. And so at the moment, anything that's different between 2 animal houses is blamed on the microbiome. I don't think everything is dependent on the microbiome, but there are some amazing data out that says that the microbiome affects lots of disease phenotypes. And to start to address that, we've developed some notobiotic capability. So we have some germ-free mice that are completely free of any microbiome, and we use these to repopulate with selective microbiome, or sometimes just to introduce a disease model, such as a genetically altered model. We can rederive that into a germ-free condition and see what effect having no microbiome at all has on the progression of the disease, and therefore whether there's a potential microbiome genetics interaction for it. These are quite expensive studies to do, so you need a real killer study for it, but they were really pertinent for what the role of the microbiome is on disease and largely disease progression. And finally, this is just a quick cheeky shout out, is that the Maryland Centre is a national facility, and we have a community called which is called Genome Editing Mice for Medicine. It's just launched for the next 2 or 3 months, and well, until July, that's the deadline. And if you have what this call does is it tries to improve those models. It tries to make the models that we're using, or it tries to generate models that are really important clinically and that are really relevant to human disease models. And the applications are review read, but you can apply for an application. It needs to be clinically relevant, improving preclinical models for therapies. You need to have shown that there is some background work done, that you're not just coming straight from a mouse, that you've taken your research as far as you can without using a mouse, but you can apply for it and they become community lines. So just in summary, I hope I've given you a little bit of a taster of thinking about new ideas for bridging the gap between human and mouse. And I say human and mouse here, but I really mean human and any model, for any model organism, that will enable us to do some much deeper experimental work. Thank you.
Okay, so maybe Carlo, Michal, Sara, if you wouldn't mind staying there, and Michal, we'll come to the front, and we can take a few questions before the lunch break. So we have a microphone if anyone has any questions for any of the speakers this morning. Maybe I will. Oh, no.
That's fine. Hi, is this working? I had a question for Carlo. So the AAV therapies seem very promising in the mouse, and I was wondering if you could comment on maybe any promising clinical trials going on in human. And I guess a follow-on question is, would they work in mitochondrial DNA? Because it obviously involves nucleic acid, so is there anything there, or is it only nuclear?
Okay. There are a few examples of gene therapy, AAV-based gene therapy for mitochondrial diseases, but AAVs are used nowadays in tens of clinical trials for other genetic conditions. So as for mitochondrial diseases, there are just a couple of examples. One has been mentioned by Michal as well, which is a gene therapy for LHON. And the other one is for Friedreich's ataxia. But these are the only two examples for mitochondrial diseases. In principle, they should work, especially for nuclear mutations in nuclear genes. I let Michal to better comment on approaches for mitochondrial DNA, but in principle, what you cannot do with mitochondria is to get nucleic acids in. But as long as the protein is produced outside the mitochondria and then imported inside, then it should be fine. So obviously, as for all AAV-based gene therapies, there may be problems, mostly related to the immune response to the viral capsid or to the expressed gene. But this is not specific to mitochondrial diseases. So the bottom line is that it should work. The problem with mitochondrial diseases is that they are extremely complex. So target multiple systems is challenging. targeting the brain is challenging. What do we mean by brain? Our brain is a very complex organ, and if we need to target billions of cells in the brain, it can be very difficult.
Are you waiting for my comment? I can. So it's no different. As long as we can deliver protein to a target tissue, and this protein will go into mitochondria, do the job of correcting mutation by base editing, for example, or cutting DNA, which is pathogenic, that should be fine. But then, at the moment, there's no clinical trials related to that. So there's a company called Precision Biosciences. They had a program targeting one of the most common mitochondrial DNA mutation, 3243, with their meganucleases, but this program stopped almost a year ago for some reason. And then within LifeArc Center for Rare Mitochondrial Disease, we progressing a experimental therapy approach with intramuscular delivery of these nucleases to test efficacy and safety. But we are on the paperwork stage, so still watch this space.
Any other questions?
Hello. I guess this is a kind of general question, and it'd be interesting to hear all of your thoughts on this. So there are so many exciting technological kind of possibilities for generating new mouse models for mitochondrial disease, but given the clinical and genetic heterogeneity of mitochondrial diseases, what are your thoughts on how we should prioritize, or what we should prioritize in terms of mouse models for studying mitochondrial disease coming forwards?
Well, if I start, I know how I would prioritise from a clinical point of view, because I would prioritise those diseases that are most clinically relevant to patients, and they're both in the mitochondrial genome but also in the nuclear genome. So, for instance, the mitochondrial genome, Carlo's just mentioned, or Michal's just mentioned the 3243 point mutation, which is the most common really that we see in our patients, and is associated with a sort of whole spectrum of problems, including very severe problems like stroke-like episodes, and that's the one that we probably, if we could choose one, we would treat. And so developing a model for that, so we can understand the disease better and then use that model to test new therapies would be a priority for me. On the nuclear side, it would be probably something like POLG, because that's the most common nuclear gene that we see, particularly in adults, and it's associated with a very severe phenotypes, particularly in children. But I don't know whether others would have alternatives.
No, I made just the comment that the other criteria we can keep into account is feasibility, because not all the loci in the genome are equally targetable.
I guess I would say a good description of what you're expecting from the model, because you can produce an absolutely great genetic copy, but it might not phenocopy. So I think it's worth having a deep think about whether you're going to be able to measure what you want to measure from that strain. Oh, sorry.
So it's kind of related to this discussion on the feasibility of genetic therapies for mitochondrial disease, given the complexity of the genetics. And I was interested in the mitophagy activation mechanism as an exemplar of a downstream mechanism that could actually be applicable across a broader clinical presentation and heterogeneity from a phenotypical point of view. So I was wondering whether in your experiment where you looked at, you demonstrated mitophagy activation in the cells, how does that translate, how does that correlate with the functional response in the skeletal example, and whether you've looked how would that translate then clinically in terms of a therapeutic effect, I guess, from mechanistically translating into clinical outcome?
It's a complex question. I wanted to make a few comments on this. So first of all, we focused on one specific model. So that needs to be expanded to other models to make sure that we are really targeting a common pathway that can eventually be exploited to treat different diseases. In terms of translatability, I think it's pretty high. That's a natural compound. It has been used for quite a long time. Not much in terms of side effects or toxic effects have been described so far. I have also to say that, as I mentioned in my presentation, the recovery we observed is partial. It's very partial. So we are not going to cure mitochondrial patients with urolithin or probably other similar compounds, but they can still contribute to the final effect. So what was the other thing you asked for?
I was just wondering, how does that activation of mitophagy correlates in your experiment to the functional, whether you've looked at that correlation?
Yeah. Yeah, so there is clearly a correlation, because when you activate mitophagy, mice run more. On the mechanism, I'm not entirely sure, because that it's not a cause-effect relationship. There may how urolithin activates mitophagy is still controversial, and we have some data saying that the activation of mitophagy is actually indirect overall. So I can't really answer your question now, but hopefully next time I will have better insight.
Okay, any other questions? I suppose I have one question for Sara. So there's obviously a lot on the use of animal models in research at the moment in the literature and in the media, particularly with sort of the pushback against using animal models, particularly with the rise of the use of various in vitro systems and organ on a chip and organoids. Where do you see the use of animal models being in the next 5, 10 or 15 years when you look forwards, and where do they fit in the landscape of preclinical models?
So I think just to, I wonder if you would have known that there was a paper out by the government that about phasing out of animals in experimentation, and I would just say that all of us who work in it, actually, that's the aim. We've always thought that animals will be replaced sooner or later. The issue is as to how mature the technologies that do replace animals, and some of them are quite mature for some questions, others are not very mature for other questions. And when you look at something like mitochondrial diseases, I went to a really interesting talk by a presentation from a pharmaceutical company who presented some really nice data from an in vitro liver damage model, which is excellent and being used as a first triage for a lot of toxicity measurements nowadays. And then they showed the other 1,000 parameters that would need to be developed in order for them to replace animals. So we're a long way from replacing them on a safety toxicology, but also a fair way from replacing them in terms of discovery science and in terms of clinical science. And most people think you're never going to replace a singular model, a singular disease model, with one single replacement model. So there's not going to be an organ on a chip that's going to give you the answer to a mitochondrial disease, because you're going to have maybe 50 different organs on different chips interlinked with other different organs. So we're quite a long way off of that. But what I do see is that we're doing much less work where we're looking at single genes in the context of a normal environment. So we're seeing much more demand from the community to look at complex models. So genes where there are multiple genes, where there are environmental challenges, as well as challenges such as age, as well as food, as well as inhalation, etc. Everything that you might imagine that would be part of a human disease phenotype. So I think where we're really moving to is less animals. We can generate knockouts in the past would take you hundreds and hundreds of animals. Now we can electroporate 30 embryos and get a knockout. So it just shows you that the scale of what we need to do in terms of animal numbers is reducing, but complexity is really increasing. And from the Maryline Centre in the last kind of 5 or 10 years, we're about at 3/5 of our animal numbers, but about half as much again of the number of experimental procedures we're doing. So we're doing much more, we're gathering much more data from much smaller number of animals, but those animals are more closely aligned with the human patient groups, and they're much more complex animal models, which is probably where the progression will continue.
Okay, thank you. Any other questions? So if there are no further questions, I'd just like to thank the speakers once more.
Welcome back for today's second session that is about diagnostics and counselling. The first speaker is Dr. Áine Moylett that is going to talk about approaches to NHS genetic diagnostics for primary mitochondrial diseases. Dr. Moylett is a translational clinical scientist working with the Genomics Hub Laboratory and UCL.
So today I'm going to go over approaches to NHS testing in the genetics lab for primary mitochondrial diseases. So briefly, just it's been touched upon this morning, but about the highly specialised service. So in 2007, along with Newcastle and Oxford, we became a nationally funded service providing this highly specialist care for rare mitochondrial diseases. And the specialised service here in London is headed up by Professor Hanna. And briefly, the service outlines that we have weekly mitochondrial disease clinics here at NHN. We have weekly MDT meetings, so this involves clinicians, clinical nurse specialists, physiotherapists, research fellows, admin, histopathology, biochemistry, and genetics. And we also have annual HSSS meetings attended by all the 3 centres. And so there's regular communication. What was it? Can you hear me okay? I can move this one. What is this? Is that a bit better? Sorry. And so there's a lot of communication and collaboration between our clinicians and genetics between these 3 centres as well, in order to provide this highly specialist service. So briefly, just going to go over the genetics of mitochondrial diseases, but they've been covered quite well in depth by this morning's session. But they can be caused by variants in nuclear DNA. They can be caused by variants in the mitochondrial DNA. But another important consideration is that variants in nuclear DNA that encode for genes that are responsible for maintaining and replicating the mitochondrial genome will then lead to secondary mutations in the mitochondrial genome that we need to be able to detect. And finally, we have the other element to consider, which is mitochondrial DNA heteroplasmy, in which there can be different populations of the mitochondrial genome within each mitochondrion, but also this can vary between different cell types. And so a bit more just about heteroplasmy. So as I said, the majority of pathogenic mitochondrial DNA variants are heteroplasmic, but not all. And there can be temporal shifts in heteroplasmy. So it's known that the levels of some pathogenic mitochondrial variants, such as the most common variant 3243, are known to decline in blood with age. And so when it comes to genetic testing, we need to consider heteroplasmy and the tissue types that we're looking at. So as Rob said earlier, mitochondrial variants tend to be selected against in dividing tissues blood, which is probably the most routinely tested sample type in genetics. But then mitochondrial variant heteroplasmy, they tend to clonally expand in post-mitotic tissues such as muscles. That's why we often do testing in muscle samples. And so as a result of this change in temporal shifts and different levels of heteroplasmy, we often need to sequence multiple tissue types to get a good understanding of the clinical picture and the genetic heterogeneity within our patients. So just considerations as clinicians that the factors you need to take into account in terms of testing for mitochondrial diseases are the age of your patient, the tissue type, and also, as we stressed earlier, that mitochondrial DNA variants are maternally inherited, and so we need to think about testing the mother and potentially the age of the mother, whether if the variant found is potentially de novo or not. So this is just a schematic of the mitochondrial genome. So it's a circular genome, about 16.5 kilobases in size. And we see lots of different types of variants that are disease-causing. So we have single nucleotide variants, insertion deletions, single deletions, which are deletions of the same size, or single duplications. Then, as we touched upon earlier, multiple deletions. So the mitochondrial genome can have deletions of different sizes throughout it. And we also need to be able to detect other large-scale rearrangements, and also there's mitochondrial DNA depletion. And so we need to be able to identify all of these different types of variants within diagnostics. And so briefly, there's the Genomics National Test Directory. So this is a comprehensive resource that's been developed by NHS England that outlines all the genetic tests that are commissioned within the NHS. So this is just a screenshot. So we have our different tests available, which are known by their specific R code. And they also, it provides further detail about the technology used for these different tests and the eligibility criteria. But I just wanted to highlight that there are plans from NHS England to digitise this. So there'll be communication around this, I think, coming soon, but I just wanted to make you aware that that is happening. So that was the introduction, and now I'll just go into kind of the testing that we do within the genetics lab. So starting with testing of nuclear DNA variants, so one of the first line tests that we do is NGS, so whole exome sequencing panels. So this is a targeted approach that sequences the protein coding region of the genome, the exomes, with a plus or minus 20 base pairs, so includes a little bit of the intronic regions as well. And so because of this, you can't detect large-scale rearrangements and copy number variants like you can with whole genome sequencing because you lack the coverage. But it's a good kind of first-line test. And so there's two mitochondrial disease panels that are available. So there's the smaller R352, the mitochondrial DNA maintenance disorder, which encompasses 25 genes. And then there's the larger R63, which is the possible mitochondrial disorders panel, which encompasses 328 genes. And these tests have a 42-day turnaround time. The next test I'm going to discuss is targeted testing of mitochondrial DNA variants. So there are the three most common mitochondrial DNA mutations, so the 3243, 8344 and 8993. And so the presence of these variants would introduce a restriction enzyme digest, and the assay that we use is a fluorescence-based assay that allows us to quantify the mutation load of these different variants. And as I said, these are the three most common mitochondrial DNA variants. And this is an example of what the data looks like. So it's what a positive sample looks like for each of the three mutations. So the wild type is highlighted in the grey box, and the mutant, the presence of the mutant variant is in the pink box. And we're able to use the ratio between the wild type and the mutant peak to determine the levels of heteroplasmy. And so this assay is like a useful first-line test, like a first-line screen in patients with mitochondrial disease, but also in patients where mitochondrial disease is only part of the differential diagnosis, and therefore this can be a quicker and easier test, negating the need for lengthier, longer, or more expensive tests such as NGS or whole genome sequencing. So next I'm going to go into our next generation sequencing of the mitochondrial genome. So this is the R300 test. And so this is done, as say, by next generation sequencing. So the library uses a long PCR template in which we amplify the whole 16.5 kb mitochondrial genome in a single fragment, which goes on for Illumina sequencing on the MiSeq. And we have an in-house pipeline in which we're able to detect variants at 1% if we have a read depth of above 500x. And so the mitochondrial genome, NGS, allows us to detect SNVs and indels, single deletions and duplications, multiple deletions, and other complex rearrangements. And we then would confirm findings by long PCR. And so, I say this is just another example of the data that we get out. So our pipeline will give us a list of filtered variants. So this is single nucleotide variants or small indels, and it will exclude kind of all background cause or variants that are very common in the healthy population. But it would also highlight any known mutations or variants that would need potential investigation. And so with variants that are pulled through the pipeline, we have a 5-class system of variant classification. So we have, number 1, not pathogenic or of no clinical significance. Class 2s that are likely not pathogenic or of little clinical significance, and these 2 classes wouldn't be included in the report. We then have variants of uncertain significance. So these tend to be variants that would need further work to determine if they're pathogenic, such as testing other tissue samples, testing other family members, or potential biochemistry or histopathology investigations. And so we'd only tend to put these on the reports if there's subsequent work that could be done to upgrade the variant. And then we have our likely pathogenics and our pathogenic variants that have, say, previously been described that are well known in the literature. So this is an example of a structural variant. So this is a single deletion. So this is the output that we get from the NGS. So the Excel spreadsheet, we have the region highlighted in red. So that tells us that there's lower coverage in this region. We then look at the sequencing data and our aligned BAMs, and we can see that there's this significant drop in coverage here. And then we can also use kind of other tools to help visualize this. So this is a mitosol output, and it's the presence of this blue line is telling us, it plots the breakpoints for us and lets us know that this is a single deletion. So that's just an example of a structural variant that we would see with our NGS. And what I kind of really wanted to focus on today is kind of maybe the limitations or things to consider as clinicians when you're ordering mitochondrial DNA NGS testing. And it's particularly about the importance of sample type and the age of your patient. And so say blood is, say, the most commonly tested sample type that we first receive. And say if you have a patient who's under 20 years old, the types of variation you'd be able to detect from this sample in a patient of this age, you'd be able to detect single nucleotide variants, duplications, and single deletions, but you wouldn't be able to detect multiple deletions. If you have a patient that's over 20 and you're looking at a blood sample, you would be able to detect single nucleotide variants and duplications, but you wouldn't be able to detect single deletions and multiple deletions. It's said that these multiple deletions tend to be selected against with age. But I want to highlight that just because something isn't being able to be detected with this methodology doesn't mean that it's not present. And if you have kind of any, if you're unsure about anything in terms of what your patient's clinical features are or the test that you're requesting, I'd really recommend contacting your HSS, speaking to the clinicians and the clinical scientists to seek advice, because we understand that this isn't, it's not always obvious. And this is why there often is the need to test another sample type. And so say muscle biopsies then tend to be the next tends to be routinely the more sought after tissue. So say if you have a patient that's under 20 years old who's had a muscle biopsy, you'd be able to detect SNVs, duplications, and single deletions. You wouldn't be able to detect multiple deletions because they tend to appear over time. So that will always be the same in your patients under 20. But if you have a patient over 20 years old, you'd be able to detect all of your single nucleotide variants, duplications, single deletions, and multiple deletions. So it's the idea that just because you're not able to see something in your first-line test doesn't mean that it's not there. And that's just something that I wanted to stress to clinicians when thinking about ordering tests. So now I'm going to go on to whole genome sequencing as kind of the next technology that's routinely requested for it's kind of like more the frontline test now. It's becoming more frontline test in genetics. And say you say they're usually taken from a blood sample, whole genome sequencing is performed and your reads are aligned to your reference genomes, and say we apply these virtual panels that can allow us to detect different variants in nuclear DNA, so our single nucleotide variants, insertion deletions, and our copy number variants. But also importantly, with whole genome sequencing, you're able to sequence both the mitochondrial genome and the nuclear genome concurrently. And at the moment, we're able to detect single nucleotide variants within the mitochondrial genome over 5% heteroplasmy. And so there are now several whole genome sequencing panels available that include mitochondrial DNA targets. And this is an exhaustive list, but I just wanted to highlight that these targets are now being included. And so what to consider when you're thinking about ordering whole genome sequencing or your mitochondrial DNA NGS. So if you've ordered whole genome sequencing, do you also need to have the R300 mitochondrial DNA NGS testing? And so in-house, the WGS pipeline has been validated to detect over 5% heteroplasmy. So within Queen Square, we did an audit of all the samples that had whole genome sequencing and R300, and the whole genome sequencing is sufficient to detect it at this level. And there will be wider communication from NHS England that will be communicated more nationally. But I just wanted to highlight that, think about test ordering and efficiency. If you've ordered whole genome sequencing, if your patient is over 20 years old, depending on the phenotype, whole genome sequencing should be able to encompass what you need, unless you think you definitely need to include a mitochondrial DNA structural variant. And again, this is where I emphasise that I think if you're unsure, it's contact your highly specialised service to seek advice about what the most appropriate testing is for your patient. So that's kind of a whistle-stop tour of all of the genetic testing that we do within the highly specialised service here. And I just wanted to go through a couple of case studies to kind of give a broad picture of the different types of testing that we have available. So in our first case study, we had a 66-year-old female presenting with a 20-year history of CPEO, bilateral ptosis, and dysphagia, so strongly suggestive of mitochondrial disease. The patient went to have the R352, so the whole exome sequencing panel, the mitochondrial maintenance disorder, and a variant of uncertain significance was found in the twinkle gene. So this gene is important in maintaining the mitochondrial genome, and so this variant was reported, and the patient went on to have a muscle biopsy, and we did mitochondrial DNA sequencing, in which we detected multiple deletions within the mitochondrial genome. And so that's illustrated here, that they have these deletions of lots of different sizes, showing that the mitochondrial genome isn't being maintained and replicated well. And so this added further evidence to the weight of the variant of uncertain significance, and the variant was upgraded to likely pathogenic, and this patient received a diagnosis. And my next case study, we had a 12-year-old who was presenting with short stature, delayed puberty, ptosis, CPEO, and retinal dystrophy, and it was queried whether they had Cairns-Sayre syndrome. So initially we did R300, so the mitochondrial genome NGS testing, and we did the analysis on a blood sample, and also confirmed this with the long PCR. So what we have here, the stars are our patient sample. This is a negative or a normal patient, and a positive patient who does have a mitochondrial deletion. And you can see that in our blood sample from our patient, it looks the same as our negative control. So this is a blood sample. But then we went on to do sequencing of a muscle sample. And as you can see, so here's the coverage plot. There's the presence of a single deletion. And we have here, again, the confirmation using the long PCR. So this is our blood sample. That looks normal to our controls, but in our muscle sample, you see that there's the presence of these two different species, indicating that there's a deletion there as well. And so it just highlights the importance of testing different tissue types. And then finally, a 12-year-old female presenting with developmental delay, short stature, new onset of seizures, and persistently raised lactate. So the investigation that was done, so concurrently we did R300, and then they were also having a trio whole genome sequencing with the R98 panel, which is the inborn errors of metabolism. And so because we had both WGS and R300 going on at the same time, the mitochondrial genome sequencing came back, and the patient was found to have 3243 variant at 65%. But because the trio whole genome sequencing was done at the same time, we were able to see the level of the variant was present in the mother as well, but at a lower level of 3.7%. So then we went on to test in other tissues. So we had urine available, and it found that the proband had a higher level of the variant. The heteroplasmy level was 87% in the urine tissue, but 14% in the mum. And so being able to test these two different tissue types enabled us to get a diagnosis for the patient, but also inform the family so that they could have appropriate counselling. that's everything for me. So thank you to everyone at the Neurogenetics Lab, the clinical team at the highly specialised service, and our collaborators in neuropathology and the neurometabolic lab, and the other 2 centres as well, and all the team at GOSH. Thank you.
Hi, thank you. I'm just going to talk about one element of biochemical diagnostics. I think we get a lot of requests, especially in the muscle biopsy testing that we do for biochemistry disease, and that's to support variant grading of mitochondrial disease. So as was alluded to before, it's really trying to build the number of points you can get to change your variant grading, and we can provide functional biochemistry evidence. I think there's still a use now where we can provide rapid biochemical diagnosis to support, for example, critically unwell infants in sort of end-of-life discussions or redirection of care sort of discussions, and providing the lab support for that. Just to explain about the neurometabolic unit, we've been doing biochemical diagnostics for about 30 years with a very young Simon Heels, who was a Puterus Fellow, who translated the spectrophotometric kinetic enzyme assays to an NHS hospital lab at the National. And the service has evolved, and the neurometabolic unit has evolved, and we are now a UCAS accredited test that provides more than just biochemical diagnostics now. And another thing to sort of stress about diagnostic testing, especially from a biochemical point of view, is the fact that we do lab testing and we do provide results and numerical results. But really for diagnostic testing, you're trying to build a quality management system about your measurement. And right now we have the international standards of medical laboratories, and these are requirements really to provide that assurance and that confidence in the number you're generating. So that's really the element of diagnostic testing, and the requirements for translational testing when we are moving things across from the research space to the diagnostic space. So back to this variant grading support that we can do. One element of diagnostic testing that's really key for biochemistry is the sample requirements and the sample handling requirements as well. So it's really important to ensure that for the mitochondrial testing we do in the lab, we do it in skeletal muscle majority from a UCAS accredited point of view, and we need to ensure we have enough muscle to do our testing in, that it's handled appropriately, and also within the laboratory we've got very locked down ways of preparing the muscle. We make a muscle homogenate, and we have everything's very set up. We've shown and we've identified that you can have artifacts if you don't handle the samples correctly for our measures. Another point is that we need to make sure we have sufficient mitochondria in the muscle biopsy. So if we have a muscle biopsy taken, we always measure the citrate synthase activity, and that's a marker of mitochondrial enrichment in the muscle homogenate that we're going to further do analysis on. And so all our respiratory enzyme activities are expressed as a ratio to this, so that we can ensure that we correct for the amount of mitochondria in the sample. And this improves the diagnostic testing for the biochemistry. So what do we test? So we do functional testing in the lab. So we're looking at biochemistry. So that's reactions, that's chemical reactions. And for enzymology, it's enzyme catalyzed reactions. So we're looking at the electron transfer activity of the respiratory chain. And we do this from a UCAS accredited point of view in the single enzymes in complex 1 and 4, and also for complex 5 as well. And to explain this image of a gel, this is our complex 5 assay, and it's a qualitative assay. So it's from a diagnostic perspective, we can only really report it in a binary way, detected or not detected. But you can sort of see that there are, you can identify deficiencies by this particular method for complex 5 activity. For our enzyme assays, we report that numerically, so we report that against a reference interval. And when it comes to variant grading, I think we've been very good at using this assay to identify combined oxidation phosphorylation deficiencies, so like translational type deficiencies, and that's been helpful in that sort of area of variant grading. We can also look at the mitochondrial complexes, the fully assembled complexes, and we do this with blue native gel electrophoresis, and this really supports the kinetic enzyme assays that we perform. So as well as finding a deficiency in the kinetic enzyme assays, we can then go on to do the blue native gel electrophoresis to look at the actual assembled complexes and identify that maybe there'll be a deficiency in the assembly that's causing the low kinetic activity. So we pair our measurements all together and to provide that functional evidence. In terms of ubiquinone biosynthesis genes, these are another area where we could use our enzyme assays to support the investigation of this. So we provide a linked complex 2-3 enzyme assay, and that requires the requirements of complex 2, complex 3, and also ubiquinone as well, where we can measure ubiquinone in itself in muscle tissue. So again, providing a combination of measurements, we can, for a ubiquinone deficiency, primary ubiquinone deficiency, we can identify a loss of complex 2-3 activity, and then also supportively a loss of muscle ubiquinone, which is the cause of the low 2-3 activity. And we quite often find a secondary loss of complex 4 activity in CoQ deficiencies. In addition to muscle, we can measure ubiquinone in white cells, and that's been helpful for looking at particular genes, especially ones that are related to adding the 10 isoprono groups to the head of the CoQ molecule, because we measure CoQ10 in itself. And genes like PDSS1 and 2, we've been really good at with white cells to identify a deficiency and evidence that way. And I think the best use of our test is really bringing it all together. So it's sort of like a functional profiling way of looking at it. So this is an example of iron-sulfur biosynthesis genes, where you can see that in complex 1, 2, and 3, they all require iron-sulfur clusters to have their to have their enzyme reactions working. And with iron-sulfur biosynthesis deficiency, you can see that you have a loss of complex 1, 2, and 2,3. And then pairing that with our ubiquinone analysis, you can see that that ubiquinone level isn't particularly low. So it doesn't really support that that's a cause of the low 2,3. But I think what's really crucial here is that we get a normal complex 4. And this is where the sample requirements and how you handle the sample, and you don't have any artifactual losses in enzyme activity, is really important. The fact you've got normal complex 4 activity and that doesn't have an iron-sulfur cluster present really supports overall this finding. And another element of support is that complex 1 has the most iron-sulfur clusters there, and we find an absent complex 1 band. So this sort of bringing all our tests together, I think this is how we use our biochemical diagnostic methods to support variant grading and provide the extra points to bump up the grading. But there are limitations to these approaches, and I think that that's really important to be aware of, and especially we're aware of it from a diagnostic perspective, especially because we need to know what the intended uses of our assays are. And spectrophotometric enzyme assays, we're measuring these enzymes at Vmax, so that means that we're giving the enzyme everything it needs and to provide the maximum velocity of the reaction. And that might miss some more subtle deficiencies, or KM variants, for example. Another limitation for all our muscle analysis is that we use whole muscle homogenate. So it's different from muscle fibers testing, like histology, you're looking at individual fibers and the patterns of that. We're looking at an average of all mitochondria when we do our functional testing, so this can have a limitation in itself. And then when you look at electrophoresis, it's a qualitative inspection of the fully assembled complexes. So I think this is also an element that could be improved on. And then ultimately we're looking at muscle biopsy, and not all patients wish to have a muscle biopsy to do further functional testing. So this is an invasive specimen that we need to kind of think about how we can move away from it. And I think we are trying to address these limitations we have. And we've had, our lab already have mass spectrometers in our lab, but we've had investment, and we're bringing mass spectrometry into the mitochondrial diagnostic testing as well. We've had investment from specific staffing for mass spectrometrists, and also we have a hybrid geneticist translational biochemist, because the focus is functional testing on variant grading. And also we've had from NHR funding through Simon Pope's grant of metabolic profiling in other disorders that we help with, this is neurotransmitter disorders. So we've already got that sort of experience in metabolic profiling. And bringing that all together to the mitochondrial space, we were able to set up a method for cardiolipin analysis, and I know Michal mentioned about this. And so we were able to provide the functional support by cardiolipin measurements in a variety of sample types to prove these variants in the MT1 gene for cardiolipin. But because we're a translational laboratory as well, but we're ultimately a diagnostic laboratory. So in addition to setting up the test and providing the measurement, we also want to provide that quality management system around that test to be able to get it to UCAT accreditation. And from doing so, we're able to provide the HSS Bath syndrome service of biochemical testing for Bath syndrome, and involve sort of like a developmental variant grading assay into actual laboratory service to support this Bath syndrome service. And I think we have other tests in development. So in the same approach of trying to set up a test, but also bring it all the way to UCAS accreditation, I think we're trying to do it with other ways because there are limitations to the assays we currently provide, but there's also a whole lot of genetic information that really needs a lot of functional work to solve, and ultimately trying to do it in a sample type that is less invasive than a muscle biopsy. So targeted proteomics, we started a partnership with a UCL spin-out company called Guildford Street Laboratories, and they provide us with a lot of support because they're specialists in proteomic analysis. But we have started with targeted proteomics, and this is essentially a way of measuring proteins, quantitative ways of measuring proteins using mass spectrometers. And because we are a diagnostic lab with the focus of getting accreditation, in addition to providing the technical abilities to measure these particular proteins, this is an example of NDAF-V1, which is a mitochondrial complex 1 subunit. We're also working at the same time to think about validation, ultimately to trying to get a test into the NHS as an accredited test. And so in tandem, we're sort of thinking about validation, thinking about what requirements are needed, and also doing the experimental work, which Rob has been doing in our lab. So this is something that we're trying to develop, and we're trying to speed up the pipeline because with variant grading, the functional work can take a long time to do all the validation to provide that level of evidence. So if we can speed that up, that's going to be really helpful. And I think something that else that's really important to pair with it is oxygen electrode studies. So this is essentially looking at oxygen consumption at cytochrome oxidase. And if you look at the panel on the left, you can see there's a whole series of metabolic pathways that all converge ultimately to reduce oxygen to water as a terminal electron acceptor at cytochrome oxidase. So this really is an ability to look at integrated mitochondrial function, so fluxomics. And if you feed in substrates at different elements of the pathway, you can actually see through an oxygen consumption measure whether or not there is a defect in that particular pathway. So you're looking at fluxomics. So this is moving away from looking at enzymes at Vmax, looking at enzymes in a physiological system, and this is also in a patient sample. And I think there's real potential to use oxygen electrode studies to really investigate VOSs. And in addition to looking at fluxomics, you can actually interrogate the integrity of the inner mitochondrial membrane. And as Rob alluded to before, the oxidative phosphorylation process is the transfer of electrons at the respiratory chain, but also the proton pumping across complex 1, 3, and 4 into the intermembrane space. And so because of the inner mitochondrial membrane is impermeable, you get the build-up, and then you need ATP synthase to release that, to use that to harness through the proton motive force. And if your inner mitochondrial membrane is leaky, you don't really get this coupling of the mitochondrial, coupling of the electron transfer and the oxidative phosphorylation. So you can add in substrates to really test that integrity of the inner mitochondrial membrane and provide a respiratory control ratio value. And we have an oxygen electrode in our lab in the form of the Ouroboros. And in fact, 30 years ago, we were doing oxygen electrode studies in muscle biopsies, mega biopsies about the size of your thumb to purify mitochondria. So we were able to do it diagnostically back then, but thinking about how you would provide that in a modern NHS in a resource sort of limited environment is kind of challenging, but we've had success in measuring oxygen consumption in white cells and cultured cells. And I think the next stage in terms of focusing on getting accreditation, providing a laboratory service, is thinking about how you would maintain sample integrity in a less invasive sample to make these measurements useful. And another element that we've sort of had development in is mitochondrial supercomplex analysis. So all my images before have shown the respiratory chain enzymes individually as separate enzymes, but actually they form together in supercomplexes. And you can study these supercomplexes by blue native gel electrophoresis, but we've also done some validation work to confirm that these supercomplexes we measure, can contain the appropriate enzymes and done in-gel activity stains. So this is a technique that we've got technically validated or technically available, but I guess the next stage is the clinical validation. Will it be useful? Is it going to be useful in terms of variant grading or some sort of diagnostic test? And that's the extra bit of work that we need to do to really focus it on diagnostics. And I think because we're talking about biochemistry, separate from genetics, you can do repeat testing for biochemistry because there's going to be changes in a patient sample. So what are the biochemical tests we could do for monitoring patients with primary mitochondrial disease? So we measure, I mentioned white cell ubiquinone, we measure it for diagnostic purposes, but actually there's evidence that it's useful to monitor supplementation. So we can show that we increase levels from oral supplementation, we increase levels within the white cells which you can measure, and then the white cell measurements correlate with muscle. So there's the potential for using white cell ubiquinone monitoring as for primary mitochondrial disease. And also another test is blood glutathione, which again we have already measured in the past, and we've shown previously that there's a potential for using it in mitochondrial disease in muscle biopsies, and it's still got interest nowadays with clinical trials that are going on using N-acetylcysteine to increase glutathione levels. So there's possibly a role for monitoring blood glutathione. And again, it's like the technical measurements are there, but it's a clinical validation. Will it have a diagnostic use? And finally, I just want to acknowledge all the team in the neurometabolic unit, so our NHS healthcare scientists who are very skilled in what they do, and also the different groups that have funded us through all the development work, in addition to our core NHS funding. Thank you.
Thank you, Chiara. Hi, everyone. So the aim of what I'm talking about now is to put a little bit more of kind of clinical meat on the bones of what you've heard about already. So thinking about all these concepts like heteroplasmy, abnormal biochemistry, etc., and how they affect the conversations that we have with our patients. There's a lot of nuance here, and I think it's a really interesting area, but I would also stress what Áine said about if you have any kind of patients that you're unsure about, we're always happy to hear about these patients and how to counsel them appropriately. And I know that's true of all of the services in the UK. So I'll go over primary mitochondrial disease genetics in terms of how it affects the patients. Then we'll talk about some of the nuances of inheritance of PMDs, a bit about prenatal testing, and then whether or not we should test apparently unaffected children. So you've heard this already. We have to think about the nuclear DNA, the mitochondrial DNA, and also our maintenance defects, which are almost a kind of an in-between scenario where you're thinking about both genomes when we're talking about PMD. We know that it's a small genome, and just to give you a little bit more detail than previously, we talk about a heavy strand and a light strand. So these are just the two different DNA strands that are wound around in a double helix. And we call this the heavy strand because it's got more guanines, which have 2 carbon rings, so they're a bit heavier than pyrimidines. And it's broken up into these different genes. And so here in kind of this teal colour, I don't know what it's called, that's the mitoribosome RNA. Then we've got these little black lines, which are tRNAs, which are used for our amino acid carrying, obviously, inside our mitochondria. And then we have these different colour protein coding genes for the oxidative phosphorylation. And it's important to think about the fact that mitochondrial DNA is replicated continuously and what that means for patients. So this is independent of the cell cycle. So it's going on all the time and responding to our cellular energy demand. Defective mitochondrial DNA can be cleared to an extent by things like mitophagy, which is autophagy of our mitochondria, but also the unfitness of some cells. So if they've got a lot of mutant DNA, they might be outcompeted. But we know from what other people have talked to you about that there is less clearance of terminally differentiated cells, and so the variants can build up in those. Mitochondrial DNA itself is highly mutable, and we've talked a bit about this already, but one thing that we tend to highlight as well is that there's this existence of these non-tandem repeats. So you have a section of sequence which is in one area, and then maybe a few thousand bases later there's a very similar sequence, and that can confuse the apparatus like the polymerase that are replicating the DNA, and that causes these deletions and duplications to start to form. And that's true of patients who have primary mitochondrial disease, like a problem with Paul G or a single deletion, but we also see these effects in lots of other diseases. So in patients with different muscle diseases, we see it sometimes in common diseases like Parkinson's and with aging. So it's really important when we see these structural changes to think about whether or not these are actually disease causing. And if you're not clear about this, it's really good to get in touch, because we are working on ways of understanding these in more detail to think about what the origin of these rearrangements might be. So we've talked a bit about heteroplasmy, but just to delve in a bit more detail, heteroplasmy is normal. We know that nearly everyone will have a heteroplasmic mutation, and they're usually present at a low level. They're usually in the non-coding space, and they shoot up the older you get. So when you're over 70 or so, you get more and more of these. And when we look at big resources like UK Biobank, we find them in there. We say that pathogenic variants also are not that rare. So there was a really good study a number of years ago in Newcastle where they looked at cord blood samples of patients and found actually that quite a few patients will carry a pathogenic mitochondrial DNA mutation. And then that's been followed up by a very big study from, again, resources like UK Biobank. And so this figure of roughly 1 in 200 probably still holds true. But it's different depending on the age of the patient. So things like low levels of deleterious mutations, like 3243, might be seen in infants and absent in the blood of older people. And why I think this is worth pointing out is that there is a real risk of an incidental finding when you do whole genome sequencing. And it's not always a good idea to chase this down the whole way, because it might mean a lot for your patients. And so I think it's important that we gather more evidence in this area, and we work together as an NHS as well, that we're getting the right balance between informing our patients about risk, but also maybe not causing undue anxiety for what might be a normal phenomenon. We've talked about these temporal shifts in heteroplasmy. One thing that's useful to point out, so the best described effect of this is in 3243, which is our mid-MELAS common mutation. And this falls especially in white cells and especially in the T cell compartment. And there's a really nice resource that was developed by a scientist called Sarah Pickett in Newcastle. And in this resource, you can pop in the persons when they were born, when the sample was taken, etc. And they do mathematical calculations to kind of infer what is their real level when they were a newborn. And I think that's really useful when you think about how you're going to approach talking to the patient. We know that variants can build up over time in post-mitotic tissue, so that would be like our neurons, podocytes in the kidneys and muscle, of course, and also heart muscle. And things like deletions might actually be able to outcompete other healthy mitochondrial DNA. So if you imagine, if you've got a circle of DNA this size and suddenly it's a bit smaller, as long as the origins of replication are maintained, it might actually replicate a bit faster. So some rules of thumb with heteroplasmy and disease. We normally say that the higher the level, the more severe the disease, but this is variant specific. And we might find that we have a high level in an affected tissue. So you might find post-mortem, you have a very high level in the heart in a patient with cardiomyopathy. That is lower than when you test, say, the brain. There's also a lot of work for particular variants thinking about what role the nuclear background plays. So it's not just about the mutation that you find in the mitochondrial DNA. If you have, to a degree, maybe polygenic risk or maybe one relatively penetrant change in an important gene, this can influence the phenotype as well, we think. And there's many exceptions to pathogenic variants in general, especially with homoplasmic variants. So I've just put in some good references to that slide as well for you to look at later. Rob has mentioned this concept of the biochemical thresholds then. So mitochondria can tolerate some defective mitochondrial DNA before oxidative phosphorylation fails. And we may have said in the past this was about 60 to 90%, depending on what you read, but it's really not a hard and fast rule. It's very variant specific, it's tissue specific, and so we need to use a lot of clinical judgment in terms of how we talk about that with our patients. We know that some variants have been reported to cause disease at quite low levels, even Lee syndrome around 40 or 50% in a small baby, depending on a lot of different factors. But in general, the lower the level, usually, if it's the true level, the milder the phenotype. The things that might be influencing what's going on, well, there's been a lot of work in the past showing that our haplogroup, so the SNPs, I suppose, that are found and inherited in the maternal line around our mitochondrial DNA may influence the penetrance of a condition, especially labours. And it's part of the story, but it's not the whole thing. There's probably environmental and developmental factors. So why is it that someone with Lee disease from mitochondrial DNA gets these really bad attacks, usually when they're small, and then seems to grow out of it over time? And then there's polygenic and nuclear background things that I've alluded to. So I'm just going to talk very briefly about some nuances for nuclear DNA counselling, which I think is appropriate for this audience, and some pitfalls we see people falling into, and then we'll focus more on mitochondrial DNA. So per the most part, it's just normal Mendelian inheritance. But one thing that we see a lot of is that a nuclear encoded gene can cause a dominant disease and a recessive disease, and typically the recessive diseases are more severe. And so a scenario that we see in our clinic is that a baby has been born and has a really severe, for example, maintenance disorder and is really unwell, and they've had maybe a trio genome sequencing or something, and the parents are carrying these variants, of course. But sometimes, depending on the variant, that might predict a risk of a later onset disease in the parent. And so we need to be slightly careful there in terms of how we give the diagnosis and also how we follow these people up. We also see in other genes, so this is outside of the mitochondrial world, but if you work in mainstream, for example, clinical genetics, there's a number of genes that are linked to increased risk of familial pheochromocytomas and paragangliomas, and these are things like fumarate hydratase, which is associated with leiomyomatosis, but also the succinate dehydrogenase genes, and some of these, so these are dominant conditions, but some of them can also cause a severe early onset recessive phenotype as well. And again, that's variant specific. So if you have concerns about this, it's a good idea to consult with your local scientist. Another thing that I've seen mentioned a few times in referrals that we receive is that one common X-linked condition, PDH deficiency, caused by mutations in PDHE, females who carry this are described as not having a risk of being affected. And actually, we see lots of patients with this who are females and are affected. So just some caution is needed with that as well. So we'll switch back now to mitochondrial DNA, and I'm focusing on inheritance. So we know it's inherited in a matrilineal fashion, and that the mitochondria in the sperm are excluded from forming the embryo. But there's no recombination. So when we talk about normal nuclear DNA, we know there's lots of crossing over, etc. And so there's some differences in the DNA from the parent to the child. But because this is essentially a clonal lineage, it means that it can be very hard to differentiate during a pregnancy what the difference is between mom and baby's DNA. And I mean that in the context of doing things like free fetal DNA testing. So that's not something that we have available at the moment. And then moving on to homoplasmic pathogenic variants. Well, these are normally passed down in the same clonal lineage, and homoplasmic variants tend to stay homoplasmic in kids. And so some of these are quite common. So the aminoglycoside-induced deafness gene, the 155 mutation in ribosomal RNA, is pretty common in the population, and it's more of a pharmacogenetic variant than anything else. And so when people ask us about this, we don't normally see these patients in clinic because we don't see this big multi-system phenotype. Rather, it should be considered that you have a risk of deafness if you're exposed to these agents. And depending on what's going on with the family and how well they communicate with each other, you might just be able to discuss this with the family as opposed to starting a cascade testing approach, which will go on forever, basically. So some nuance is needed there. Other conditions such as maternally inherited cardiomyopathy, 4300HG, this is rarer in the population. Again, it has variable penetrance, but we do cascade this quite aggressively because of how severe it can be when it happens, and we want to see this in patients' notes and make sure that the cardiologists know about this. Relatively similarly, there's another very rare condition called reversible COX deficiency, which we tend to test multiple family members for. And then I just wanted to come on to Leber's. This is coming up a lot at the moment in emails that we're getting. So the main cause of Leber's, these common homoplasmic variants, are not that rare in the general population. And sometimes they're reported incidentally. The BSGM have recently written some guidance, I think, that they're going to say not to report these. But if, say, you have an unwell child who has some visual problems, you mightn't be able to determine whether or not this is incidental or part of the cause. So they do sometimes get reported with or without this guidance. And it can be tricky to know exactly what to do in this case. If the family don't have a history of blindness, it's not always that useful to do a lot of cascade testing. Depending on the family that you're talking to, it might raise an awful lot of anxiety, and the risk of having disease is pretty low if there's no history of blindness in the family. So you need to kind of take this on a case-by-case basis. If they're quite relaxed about it, I typically say to them, these are the symptoms. If you ever became unwell, you get this centralized loss of vision, and it normally comes on in late childhood, early adulthood. These are the people that you'd go to see if you had this. This is what you'd say. And you need to avoid things like smoking cigarettes, drinking too much alcohol, that kind of thing. And because there's a nice approved treatment now, it is, I think, important they know about this. But we don't have enough evidence to say that everyone should have this reported, I guess, in terms of penetrance. And that has been a decision made by BSGM as opposed to the mitochondrial labs. So at the moment, it's not normally reported incidentally. So the next thing I wanted to touch on is the bottleneck. So Rob has told you a little bit about this, and the long and the short of it is that a small number of mitochondrial DNA molecules in the primordial germ cells are the template for the next generation and divided into our oocytes. So it means that there's a small number of mitochondrial DNA seeding the next generation, and by chance, you might get a high load of heteroplasmy mutant DNA or low. And so there can be differences between mum and child. I've given some more detail on this here, but some of the important things to think about is this is a relatively random, a stochastic effect. There may be some influence from nucleoids, which Rob mentioned earlier, so the kind of clumping of mitochondrial DNA around proteins. It's an area that there's lots of research around, but there are also variant-specific factors here. So we tend not to see patients with 3243, for example, at 100%. And so we kind of infer that these are really damaging variants, and maybe oogenesis is failing or the embryos aren't succeeding or viable. We also tend to see that the single deletion isn't normally passed on. And there was some work quite a few years ago now that estimated the risk of that being transmitted at about 4%. There's probably a lot of nuance to this that we still don't know about. So we're always happy to discuss individual cases and think about might there be a slightly higher risk in your patient. We know that a single duplication in our clinic is highly heritable. It basically behaves pretty like an SNV because it's not causing this, I suppose, massive drop in dosage that you would see with the deletion. It's having more of a frame shift effect. And so if this is the case, it probably is very heritable. In the families that I've looked at, nearly everyone, apart from one patient, has received it from mum. So we're happy to discuss these, and there can be nuance when you think about rearrangements about what the risk is. Here's some good references for those. So hopefully that makes sense so far, and then we'll talk a little bit about prenatal testing for heteroplasmic mitochondrial DNA variants now. So other prenatals for nuclear encoded genes are the same as normal, but it's often important to think about, say, for example, if you've got a family with a previous diagnosis of one of these maintenance disorders, and the mom and dad are saying, well, would we be able to get, or someone else in the family, like an aunt, wants to have PGT for carrying a late onset variant. That might be very difficult because, you know, in the UK we license these technologies for severe pediatric onset disease. So I wouldn't jump in and say that would necessarily be available, and it may not be appropriate. So the kind of background to doing prenatal testing, and there's been huge amounts of work in this area, is that we think that these heteroplasmic mitochondrial DNA variants are stable and homogeneous in the womb. And so when post-mortem testing has been done of fetuses, they tend to be the same in every tissue. And so based on that, we say that if you do a CVS or an amnio, it should be representative of what's going on in the baby. So the most common sample that we receive here in Queen Square is a CVS sample. And the good thing about that is it can be done early. And we would say that the sample needs to be a normal size. So you can't send us a tiny sample because we won't be able to culture it. Because if you start culturing the cells, then you could get shifts in the heteroplasmy level, defeating the purpose. I think the second thing to say is that there's some research saying that there can be some variability depending on the mutation around the placenta. And so you want to have a big enough sample that you're getting a kind of a pooled representation of the cells in that area. The prenatal technique is best if there's a low recurrence risk, and we'll come on to this. But for a lot of variants, it's really hard to predict the phenotype from the variant load. So what I would say is try not to promise anyone a prenatal. When I see a prospective mum in clinic, I actually sit down with the scientist before I see her and think about, would we be able to do this? And what would the report look like as well? So it's best used for apparent de novo cases because these have a low recurrence risk. And so the situation is normally that you've a mum who's had a severely affected child and that baby had a really high level. And then you've tested the mum's blood, so maybe she had an R14 and it was absent, and then you followed it up by doing it with a more accredited test. And so experience would tell us that the risk here of this happening again is really low. But in order to say it's de novo, we need to think about it in a bit more detail. So normally when we talk about de novo risk of recurrence, what we're saying is that the risk is that there's gonadal, or germline rather, mosaicism. Whereas here, we're also saying that we could be missing the variant, not because it was mosaic in this lady when she was born, but because it's disappeared in certain tissues. So to say it's de novo, you need to test multiple tissues. And in general, to the best of my knowledge from reading the literature, the only time a de novo variant has been reported as recurring was when a post-mitotic tissue wasn't tested. So it depends on the situation and the family that you're talking to. If they're very risk averse, they might want to have a muscle biopsy and have this test during pregnancy, and they might not if you give them the full information. But it's about really empowering their decision as opposed to having a catch-all approach to patients. But that is a situation in which a prenatal can be useful. And then there's one or two variants that are really well described from the early 90s. So this recurrent variant that Áine mentioned earlier that can cause a maternally inherited Lee syndrome and ataxia and blindness in adults has been looked into in a lot of detail. And this is from the original paper back in 1999, but they inferred that the risk for this specific nucleotide change reaches a bit of an inflection point around 50%, and then at 75% for this nucleotide change. And so that is a really useful rule of thumb, and it's something that we think about when we're offering these tests. There are some more up-to-date papers on the risk with these specific HP6 mutations. And so that is a variant that we would offer prenatal testing for usually, as long as the mum is well counselled, and especially if she has a lower level, I guess. There is also a school of thought that this variant will skew very high or very low in mum's oocytes, although this is probably quite complex. So what about everything else? So yes, it's good for de novo, but there's lots of other things we need to think about, isn't there? So the problem here is, how do we correlate the level with the offspring's future phenotype? So we have these empiric figures where we say about 30%, if it's lower than that, there's a low risk of severe childhood onset disease. In France, in some centres, they say 40%. So, you can see that it's just largely empiric. There is some data on some things, but for a lot of rarer mutations, it's impossible to come up with this data. And then what about high risk of severe disease? Well, usually in our lab, and I think in the other NHS labs, we say over 70%, but they say 60% in some countries. We could say that there's a high risk of severe disease, but you've got this massive thing in the middle between 30 and 70%. And for a lot of mums, the most likely place that their prenatal test will land is in this 30 to 70% area. So what would you tell them to do? How would they deal with this? And it's not necessarily that we say not to have this discussion, but they really need to be incredibly well informed before they embark on this. And if it's the case that mitochondrial disease is something that they're living with and they're already pregnant, sometimes they might want to avoid this. But also we are so lucky in the UK now that we have this Specialised Reproductive Centre, which Bobby is going to talk about later. And so getting a referral in there can be really helpful in terms of dealing with these difficult cases. So they might be able to have something like PGT. A question then that we often get would be, are there any edge cases? And there definitely are. And myself and Joe, who's the GC in Oxford, were talking about this recently. So we really do worry about this and think about it a lot. And we're in constant communication. So if you had a mum with a very low level of the variant in multiple tissues, she's probably more likely to have low-level eggs. There was some work previously that said if her level of even 3243 was very low, then there's a reasonable chance that some of her eggs have a low level as well, and that's the basis of doing PGT, essentially. But we would need to think about this. Then there's also, say, a family who've just lost a child, and this does happen, with a really high level, and the mum has a moderate level, and they're now pregnant. So would you tell them they absolutely can't have a prenatal test? Maybe you would, but is that being very paternalistic? What if they said, I'd be happy to continue as long as they don't have the same level as my previous baby? So it's really tricky, and we should always discuss these cases. In general, with the single deletion, we tend not to do any tests for that, but case-by-case discussions are appropriate, I think. Coming towards the end now, the general approach that we take is for testing of offspring that we normally say not to do this. So if mom has a mutation in mitochondrial DNA, if she's not having any prenatal interventions, probably best not to test the child to protect their autonomy. But it's also the fact that we don't know what to do with that result. So say you get a result back and the baby has 50% mutation. Would you tell them to have any specific screening? Well, we don't really have an awful lot of data to support exactly what you should do with the kid. Will it cause a lot of anxiety if it's at a high level? So, sometimes some centres will do a test and the baby could have a really high level and then mom is really concerned, but we've nothing really to offer in terms of preventing a bad outcome. So we just need to be a bit nuanced there. Where a mitochondrial disease is really well known to a family, they might be a bit more relaxed. We could take more of a common sense approach. Some families are very concerned. It might be a good idea if we're not testing them to see if a local paediatrician, like a metabolic paediatrician, we've got some great colleagues in London who have been really helpful with this, might see the child intermittently and just make sure that they're getting on okay. And we are working on some guidance for this. But it's definitely something worth thinking about. I think it's also the case, though, it's different to your normal nuclear stuff. So it's not like we're doing a test for Huntington's that's completely predictive. We know this is passed on. It's not a 50-50 risk. We've had really rare exceptions, but nearly always the baby will have it at some level. So there is more nuance to it than saying, is it there and is it not there, that we need to think about. If we do a prenatal test, we tend to offer a confirmation cord blood test as well, although this isn't a hard and fast rule. And then I would say that in certain cases, it could be useful to confirm the homoplasmic variant. And I've mentioned here that cardiomyopathy variant. So we know the baby will have it, but you might as well have it on paper so that the cardiologist, who mightn't be an expert in mitochondrial DNA, for example, will be doing the appropriate follow-up. And to finish then, I just wanted to talk about a common predicament that we see in genetic counselling that we've kind of written up before and talked about, and just why it's important to think in advance before you're doing a test what it might mean for a family. So imagine we have a female and she's had testing for monogenic diabetes. So this isn't a test that's done in one of our core labs. It's a bigger test. There's lots of causes of monogenic diabetes, but we know that 3243 is on it. So imagine, and this happens all the time, that she's got the variant at 50%, and you might say, okay, that's great, we've made the diagnosis and we could do some things to optimize her management, and that is good. But we'll also say to her that there might be a risk of CNS disease in you, and that could be really awful, we're not entirely sure. We tried to think about her family history and a few things like that to infer the risk, and there's been some really nice papers as well. But we don't yet have any clear clinical guideline that we can tell someone how high their risk is. We might also be giving a de facto diagnosis to lots of other people in the family, because it's not that it's this 50-50 risk. We tend to see it kind of in everyone in general. There's rare exceptions, but this is what we normally see. And then imagine the case where she's already pregnant, and this does happen. So she's a really poor candidate for prenatal because her mutation is slap bang in the middle. You'd love to send her for PGT, but she's already pregnant now. What if she wants to have the prenatal test anyway? What can you tell her? It's a really difficult situation. And I think if you have a high suspicion that it might be mitochondrial diabetes, it's useful to say in advance, this could come with some extra things, and just give a little bit of a flag of warning that there's a risk of this. So we wrote a paper on this a few years ago, and I think it's worth thinking about if you're working in this area. So to summarize, counselling and prenatal tests are complex for mitochondrial DNA. I hope I've gotten that across. Think about mitochondrial DNA as variant specific more than just a gene panel. Avoid offering prenatal testing until you've discussed it with your local team. Wherever they are in the country, they're always keen to hear about things in advance. And you can always get in contact. So in London, we have a weekly MDT for ongoing clinical queries. We often get external people joining that about something active. And then we have a monthly complex genetics MDT. So say you've got a VUS, an incidental finding, anything like that, you're really welcome to come. And we have people from around the country and different countries as well. We have people from our genetics team, but also our neurometabolic team. So we might be able to come up with an assay that would upgrade your VUS, like Amanda was talking about as well. We've got a laboratory email and a clinical email that you can contact, and this is my email on the slide as well. So we're always really enthusiastic to hear from you. That's it. So thank you very much for listening.
Thank you. So we now have 10 minutes for questions. While we wait for some questions, I have a question for Amanda. So in the era of advanced genomic testing, what's the current added value of biochemical assays, and in which clinical scenario do they change patient management?
Okay, so I think biochemical assays, I think what we're looking at is actual reactions, functional testing of actually doing something. You can look at proteins and you can look at mRNA transcripts, and that's amounts of things, and you're inferring function from that analysis. But actually biochemistry provides you that dynamic process, so I think that's what the value of biochemistry is. I have to sell my discipline here. Yeah, and I think ultimately you have these variants, and they are good, you know, that's instructions for what's going to be made, but the biochemistry can provide the end point. So it's really essential, and I think with these genetic variants from almost like the patient-specific elements, there's more of a personalised medicine. That's why the proteomics approach of really doing that personalised testing and trying to speed up that functional biochemical evaluation is really key to that, because it can take a while to set up an assay, but if you get something, a good quality system set up, it helps. In clinical scenarios, I guess it ultimately is for genetic counselling. You know, once you've been able to grade that variant, it's adding those points and having that assurance and that quality that you have confidence in your biochemical result. That's where the biochemical testing is, and then you can have that extra confidence with that variant grading. So then we see the genetic consequences. That's the clinical management, I think, with biochemistry, and also the monitoring that I showed. You know, so that's
Thank you.
the one here.
Thank you. Hi, thank you so much. I'm Harriet. I'm a genetic counsellor at George's. So for Will, mainly, trying to prioritise the things go through my head, but we email your team a lot. So it's kind of trying to think of how to streamline that, really. So literally, I'm kind of simplifying now. You've mentioned the weekly MDT. So really, it's our prenatal settings that we come to you with questions. And would an approach sort of being, if we get a referral in where we think there's going to be a request for prenatal, that it might be easiest for everybody if we kind of attend the meeting with our questions?
Yeah, definitely. I think if you're based in the London area, and your email address is very familiar to me, so always happy to hear from you. If you are based in the area, definitely you'd be super welcome to attend, and we have our genetics lab team there as well, so they can give some extra insight. The other thing is that, you know, this issue is coming up quite a lot, and so we're meeting together with the labs from around the country in the coming weeks to try to pull together more formal kind of guidance for ourselves and more widely for this area. So we will try to develop something, but as you can see, it can be very nuanced. And so, yeah, you're definitely really welcome to attend. And it's every week, so hopefully it won't cause too much of a delay if there's an active pregnancy and stuff like that. Yeah.
There was one here.
Hi, thank you very much for that. It's mainly a question for the first speaker. What is the value of using urine for large-scale deletions? I'm just particular, I've got a patient that's got a KSS type phenotype who's 40, and so there's obviously no point testing her blood, but could we use urine rather than muscle?
So we don't test for large-scale rearrangements in urine at the moment, just as a limitation of the sample type and getting the DNA quality out of it. So in that case, I'd say advise more of a
Yeah, we'd normally get muscle on some of that age. We do sometimes make exceptions, and we try to do it. So if you email us specifically about the case, and you know if they're very muscle biopsy phobic, which is understandable because it's not very nice, we can absolutely look into it. There is a problem in terms of the amount of DNA that we get out and the quality of the assay as well. Some centres do buccal as well, which is something we'd like to expand into. The guys in Newcastle are doing that at the moment as well. And I think ultimately we do need to look at the less invasive things. Áine has done a lot of work recently about moving towards different extraction techniques, and so we're hopeful that we will be able to change these things, especially as treatments are coming around and we need to do things faster. But what I would do in any of those cases is email your clinical scientist team, and then they'll tell you how confident they are, and they're probably happy to give it a go, but they don't want to raise your expectations every time.
Okay, thank you.
My name is Dua Salman, and I'm currently working in John Walton, and I will join the Newcastle mitochondrial team in September. I have a question related to the genetic testing when we have a variant of unknown significant, especially for the population who are underrepresented. Like I did one of my research in Lebanon, and I found like in clinical picture it's going with autosomal recessive mitochondrial disease, but the patient have one pathogenic variant or likely pathogenic, the other is variant of unknown significant. And at time of publication, it was like bioinformatic analysis or in silico data analysis predicted it's pathogenic, but there is no functional test have been done. So my question is how you deal here with the variant of unknown significant if you consider the clinical picture as evidence for upgrading it, or you do functional testing, or?
So it depends on the variant. So if for patient specificity, usually to upgrade variants, there has to be kind of a known biochemical signature that we can measure. So it's not just that they clinically present that it sounds like the disease. We need to have kind of evidence that it is disease and gene specific in order to that to be considered as evidence. And then if that isn't available, then that's considered. Will mentioned our complex genetics MDT. If there's maybe potential that there could be functional work that could be done on the research side, these cases are always welcome to be discussed to see what we could do if there's other ways that outside of the NHS testing that we could potentially upgrade the variant. But it's obviously it's gene and disease specific.
I think we've had a big influx of VUSs in the coenzyme Q10 pathway, because we know increasingly that they can cause limited phenotypes like retinal disease or muscle-specific, kidney-specific, that they don't always cause the multisystemic disease. And Amanda's team have run quite a lot of samples for us in the last while and have upgraded a good number of variants, either using the blood ubiquinone level or the muscle. I think that's true, Amanda. So so yeah, we're always happy to talk about that and receive samples. And when we do our complex genetics MDTs, there'll be someone in the background doing the sums, how close you are to likely pathogenic. And so if we don't think it'll make any difference, we won't recommend doing it. But yeah, I hope that's helpful.
Any other questions?
Yeah, this is a question from a non-geneticist, so I'm a biochemist. But is there any use in looking at extracellular nucleic acids? Because there's this talk of some of these mitochondrial defects in blood cells. They're not very useful because they're basically getting rid of all their sort of bad mitochondria. And I know from looking at CSF, we get immune response activation in some of these mitochondrial disorders because you're releasing things which aren't normally exposed in the extracellular environment.
I've never thought about that, to be honest with you, but I think it's really sensible because there is some inflammatory changes in a lot of the mitochondrial diseases and the proteomic pathways. I don't know, maybe Rob or Bobby or someone would have a thought about that, but maybe it's something for tea. Okay, hi everyone. We're just going to start again. So this is our last session, and our first speaker is Renata Kabiljo. Renata is a senior bioinformatician in the Institute of Neurology, focusing on mitochondrial diseases. She works mostly on a project funded by the Lilly Foundation to boost diagnostic rates, and she's an honorary position with our GLH as well. And so she's going to talk to us a little bit about going further with the genomic diagnostics for primary mitochondrial diseases.
Thank you, Will. I will start by introducing briefly the Lilly Precision Diagnostics project, although I think that we will have a talk by Katie after this, and she will probably tell you more about that particular project. But mostly I will talk about how we make the most from next generation sequencing data, and then how we use long-read sequencing for these large-scale rearrangements in mitochondrial DNA. So Lilly Precision Diagnostic project, I'm solely funded by the Lilly Foundation to be bioinformatician on this project that aims to diagnose individuals with suspected mitochondrial disease who haven't yet received an NHS diagnosis. But apart from making this diagnosis for previously undiagnosed patients, we are really looking to accelerate adoption of these advanced genetic technologies into the NHS. So it's not just about finding diagnosis for the patients that are recruited, it's also about finding ways for our methods to go to the NHS to help more patients. The recruitment criteria, the patients can be of any age with suggestive phenotype and biochemistry, either when they have had genetic testing and either no variant has been found or a variant of uncertain significance. And we already had 150 patients recruited to the study. So the study is well underway. And on the UCL side, this is the team. We are part of Rob Pitceathly's lab. And on the bioinformatics side, we have myself and Tamara, who is a research assistant and a PhD student funded by LifeArc. And you have heard this morning from both Will and Áine, who are on the interpretation side of this project. So let's start with the first bit, how to make most of this whole genome sequencing data, how to improve. We are focusing on improving bioinformatics pipelines and reanalyzing what we already have before we embark on this new sequencing with new technologies. So this morning, when Áine finished her talk about current testing for primary mitochondrial disease in the NHS, Áine was talking about the panel-based approaches, yeah, and also the last part of Áine's talk was about whole genome sequencing, and so this is sort of where I start from, the whole genome sequencing and panel-based. So the first thing we do is to revisit clinical notes, and we can apply very easily additional panels. But more than that, there are genes that we are interested in that are not on panels. For example, there is a database, Mitocarta, that collects more than 1,000 mitochondrial genes, that some of which have never been implicated in disease, but we still want to see, could we have any pathogenic variants in these genes. So the first thing that we do is we look at more panels and our own collections of genes. So this is still panel-based. But then we also move away from the panel-based. So we have these agnostic approaches. In particular, if you have a trio recruited together, it makes it easier for us to find these variants that are de novo, that have not been inherited from either parent, but appeared in a child. Also, we can look at we have this dedicated pipelines that we wrote in-house to zoom in on variants that are either de novo or homozygous recessive or compound heterozygous, which is when one pathogenic variant has been inherited from each parent, X-linked, etc. So this is so we serve it panel-based, agnostic in parallel. All this is like without resequencing anything. We are using whole genome sequencing data that already exists. But what underlines our approach here is also using new bioinformatics tools for all of this. So I will show you in a bit an example where just replacing one bioinformatics tool caused us to diagnose many new families. So apart from the actual tools to find these variants, we are improving annotation of variants with clinically relevant information. So what does that mean? That means that for every variant, we go to many, many different resources and databases. We check the frequency in general population. We check some kind of predictions, whether that variant might be pathogenic or not. Could this be a splicing variant, etc. So we're really expanding the list of resources that we are including here. And then based on these resources, we have this filtering script that we prepared, and we are ranking these promising variants for prioritization based on our annotation. Here we hope to speed up the interpretation, so that something that we deem to be, that we computationally can put on the top of the list as potential culprit will hopefully be the causative variant. Now, one thing that I will not have time to focus on in this talk, but what we also did in a couple of cases, and it's a very important part of diagnosis, is using RNA-seq. In particular, if we have a variant of uncertain significance, for example, splicing variant, we can use RNA-seq to see the effect on translation. And we are using a pipeline to detect both outliers in expression and in splicing. So this together hopefully brings new diagnosis with existing data. Oops. So I promised you one example where just replacing one bioinformatics tool had a huge impact on diagnosis. So, as you've heard, like maybe 5 times today about this heteroplasmy, that the mitochondrial variants are different than nuclear variants, that they're not homozygous and heterozygous. They can be homoplasmic and heteroplasmic. So this confuses these variant callers, which are computer programs to find these variants. And that's why just replacing the standard bioinformatics tool with one that was dedicated to mitochondria that is very good at calling these heteroplasmies had a huge impact. So we started with a cohort of cardiomyopathy patients and Charcot-Marie Tooth patients in Genomics England, and we ran that tool on all of them, and we uncovered several new diagnoses. So you also saw similar graph before today. So, for example, 4300 has been mentioned already many times today. So there were actually a couple of families that, for some reason, in Genomics England, the diagnosis has been previously missed. So when we took that tool, we found the diagnosis, and another few variants. And then when we saw how good it is, we worked with a team in Cambridge that had some bandwidth to run it really large scale in the entire rare disease Genomics England data set. So it was Dr. Catherine Schoen and her PhD student, Jiming. And there were 51 families that suddenly had a suspected new diagnosis with a pathogenic variant in mitochondria. And this is not just the undiagnosed mitochondrial under category of mitochondrial disorders. No, we had their categories were retinal disorders, inherited neuropathies, cardiomyopathies, intellectual disability, epilepsy, etc. So, example of just changing one tool, 51 possible new diagnoses. So that was until now. We haven't sequenced anything. We just applied better tools and better method to the data that we had. And now, let's see what these new sequencing technologies can offer for diagnostics. So I was wondering whether to put this slide or not, because I see that most of you, if not all of you, are not bioinformaticians. So if there is like a little chance that not everybody knows how DNA is sequenced, then maybe these few slides will be quite helpful, but it will be just a few, so it's not going to be a long lecture on how sequencing works. So I think it's important to know that when we sequence DNA, we cannot sequence the whole molecule, which would be a whole chromosome. So we shred it and amplify and shred it in tiny, tiny little pieces. And then the big bioinformatics task is to assemble these. We sequence little pieces, and then we have to put them back together. So imagine that you're doing a jigsaw puzzle, and it's like many, many, many pieces. And we are talking about millions of pieces. And now think about if you do a jigsaw puzzle, all these pieces with the sky or the sea, or I don't know, that are left at the end, and you just simply don't know where to put them, or they fit in many places and you're just not sure. So this is the problem with whole genome sequencing, with this, we call them short reads, with what Áine was saying, that NHS sometimes is using that for whole genome sequencing. So now if we didn't find a variant, if a person is without diagnosis, maybe we didn't find that variant because that variant was on these little pieces of sky or sea that were left. We didn't know where to put them, so that's why we didn't find it. So that's why let's start by trying to assemble our picture with a bigger puzzle, with the bigger pieces. So fewer pieces, but bigger puzzles. So, for example, now if you have the same picture, but now puzzle pieces are, in this case, like 100 times bigger. So it's much easier to assemble the whole picture. So the whole genome sequencing, typically we are sequencing one segment of 150 base pairs, and here we are going to tens of thousands, depending on technology. But we can do tens of thousands sequenced in one go. Now, as has been said like many times today, the mitochondrial genome is a circular genome of 16,500 base pairs. And as I said, we can have tens of thousands nucleotides on one piece of puzzle, one read. Yeah, that means that actually I will have the entire mitochondrial genome sequenced in one go, in one molecule. So I almost don't need to assemble it. That's how long my reads are. They will sequence the entire mitochondria. So why? So of course, this long-read sequencing is good for nuclear DNA and for mitochondrial DNA, and we sequence the entire genome, so we have nuclear DNA and mitochondrial DNA sequenced with it. But I will tell you, in particular, when it comes to mitochondrial DNA, why are these structural variants difficult to resolve with whole genome sequencing? So we have, imagine that this is one wild-type mitochondrial DNA. Now, I think both Will and Áine and Rob, everybody was talking about these deletions. Yeah, you heard about mitochondrial deletions many times. So, so this is how it would look like if we had a deletion. Yeah, you see this whole purple arc is missing. Yeah. Then we noticed something that is not really described that well. We noticed that what can happen is these two molecules with the deletion fuse together. So we had something that looked like a duplicated deletion. We call it deletion dimer. And also we have duplications. Yeah, when one segment, now we have twice that purple segment here. So these are kind of structural variants that we see in patients, and it's difficult to resolve them with whole genome sequencing, with short reads, with tools that we had. So it is well known that tools, whole genome sequencing tools, tend to get confused between a deletion and a duplication. And why? Because they rely on coverage, how much DNA is there over our mitochondria. I'll show you more about how it works, and tools struggle to tell them apart. But we also heard earlier today that a single deletion, typically not inherited, but a single duplication inherited. So it's really crucial for our patients to be able to tell them which one of the two. Now, why is it so difficult? Imagine if you stretched, now you stretch this molecule, and the bottom is showing where, the bottom graph is showing like the amount coverage, the amount of DNA I have over each place along the mitochondrial genome. So it's clear that something happened here, yeah? But the genome is circular, so I don't know, has one part been duplicated or one part has been deleted? It's very hard to tell, and this is why the tools got confused. So it's something that was difficult before, and mistakes have been made frequently. So here's a case of one of our patients, 60-year-old female patient with drooping eyelids, double vision, weakness at hips and shoulders. And this coverage plot, how much DNA we have along the chromosome. Here you're looking at we linearized mitochondrial chromosome, so you can see how much DNA is over each place. So we see that it's either single deletion or single duplication, because we don't know whether it's deleted or duplicated. But now we have these long reads. So first of all, this is alignment. We call it alignment. So every line is like one molecule we sequenced. And then you will see that in some of them, you see that there is a hole, a gap, yeah? So this is the actual deletion. So this time, with my long reads, the entire deletion is captured in a single molecule. So we also developed a tool to call these structural variants, bioinformatics tools. And this tool has called, this is a deletion, I'll tell you more about it. But so this upper circle where it says wild-type empty DNA, these would be like the gray lines, and deletion, smaller molecule, would be these lines with where you see this gap of almost 8,000 nucleotide. So these are the two molecules we see easily, and our tool, we call it MitoDetective. It plots it as a single deletion. It calls it as a single deletion. But we saw something else here. So just to see whether we have some more complex structural variants, we have created, just for visualization purposes, we have created an artificial reference molecule that contains two chromosome M's fused together. So we took linearized the molecule, glued it 2 times, all just on a computer with the files. This is not lab work. It's virtually we copied the text, the sequence of the genome of chromosome M, and glued it twice, and then we aligned all these molecules that were sequenced to that reference, and then we saw something very interesting, that when aligned to this double chromosome M, yeah, we have these molecules that align twice and contain the deletion twice. And these are the deletion dimers. So we think that clinical significance of these is exactly a single deletion. Now, because we don't have that many patients yet with these kind of structural variants, we are not sure whether every patient creates these. Is there something here with significance? But it's very important. Our tool now, the output is, yes, single deletion detected, yes, presence of deletion dimers. So this is something new that we are doing. And now another patient, this coverage plot looks almost the same, different breakpoints, but this could be deletion, this could be duplication. It's a 50-year-old male with diabetes, deafness, and poor balance. So this time, when aligned to double chromosome M, you never see these deletion dimers. Why? Because you see that we have some of these some of these reads that span through the entire chromosome M once and then have an additional part. So this is a duplication. And what we are seeing here is that this duplicated molecule with two purple arcs, double purple arcs. So this is what we see here in this plot. That means that one molecule contains the entire chromosome M and then extra duplicated piece of chromosome M. And our tool called it successfully as a single duplication. And now the multiple deletions. I believe this has been mentioned also several times. Will was talking about this. The multiple deletions are a hallmark of primary mitochondrial disease maintenance disorders. But they can also be caused by aging and other disease processes, for example, neurodegeneration. So we would like to know, patients with multiple deletions, is it a real primary mitochondrial disease, and even like what kind? And so what we are doing, our tool that we call MitoDetective, the tool calls these multiple deletions. And if you see on the left-hand side, these two plots are both plots for MitoDetective from two multiple deletions patients, and the plots look very different. So Tamara is our PhD student, and her focus is looking at the structural variants in mitochondria. So she's comparing different features of these deletions and to use them as some kind of biomarker for primary mitochondrial disease. Even preliminary PC analysis will show that, yes, some patients who for whom we do believe that their deletions are caused just by aging, clustered together. Features of these deletions were similar, and some patients who have particular kind of mitochondrial disease, particular, for example, POLG mutation, they also clustered closer to each other. So this is quite promising. And we really hope to gather more patients with multiple deletions and then to develop some kind of predictive algorithm to use in diagnosis based on signature of multiple deletions. I think that's all I wanted to say today. So to summarize, I hope that you saw that we can actually improve diagnostic rates by just reanalyzing the data that we already have, whole genome sequencing data, and also that these new sequencing technologies, such as long-read sequencing, are quite useful for these large-scale rearrangements in mitochondrial DNA. Thank you.
Great, thank you very much, Renata. So next up we have Chiara Pizzamiglio. Chiara is a consultant neurologist here at the National Neurology Hospital. She has a special interest in neuromuscular genetic disorders, in particular mitochondrial diseases and myocardial and related syndromes. And she's going to talk to us a bit about clinical trials and biomarkers.
Thanks, Will. So the topic of my talk today is quite dense, but I'm conscious of time. I only have 20 minutes, and I'm the last speaker of the day. So I will mainly concentrate on these three things. So I will briefly introduce clinical trials in primary mitochondrial diseases. I will talk a bit about biomarkers and outcome measures, what we currently use, and then I will show you some results of our research on muscle MRI in primary mitochondrial diseases. So why this matters? So first, we know that mitochondrial diseases are quite heterogeneous, both clinically and also genetically. It's quite difficult to measure the progression of these diseases because in most cases, especially in adult, they are slowly progressive over time, and there has been a limit trial success. Although we have some approved treatments for primary mitochondrial diseases that are the ones that you can see on the right of the slide, most of them has been approved this year or last year, and are idebenone for Leber, nucleoside therapy for TK2, elamipretide for Barr syndrome, and Q10 for primary Q10 deficiency. But everything else is still not treatable. We don't have a cure. So there is a multitude of metabolic pathways that are implicated in mitochondrial function. And these are all potential targets for primary mitochondrial disease therapies. Some of these compounds aim to increase the mitochondrial function and have potentially broad applications across multiple mitochondrial diseases. Others are more specific to a particular molecular mechanism dependent on the underlying genetic defect. There has been a substantial increase in trial activity in mitochondrial disease over the past 10 years, and this has been driven by both the interest from industry but also a commitment from the research community. However, the pace of the increase in trial activity was not followed by the production of reliable outcome measures that are able to capture treatment responses. And this has probably contributed to the limited number of clinically approved compounds that we currently have. However, you may have heard this morning, there has been an exciting development in the treatment of mitochondrial DNA-related mitochondrial diseases. That is the development of technologies that allows the editing of the mitochondrial genome. This is a major advance because mitochondrial DNA is resistant to CRISPR and has consequently been extremely difficult to manipulate. So this does raise the possibility of genetic therapies for this group of disorders that are still under studies, though. And this is especially for mutations in the mitochondrial DNA that are the most common cause of mitochondrial diseases in adults. As you can see in this slide on the left, we can have a mitochondrial DNA elimination strategy using mitochondrial DNA nucleases. So they can selectively target and remove only the mutant mitochondrial DNA. And when this happens, the concentration of mutant mitochondrial DNA drops below the pathogenic threshold, and cells can regain a normal phenotype. While with the base editor, we can do something different. We can correct or install point mutations in the mitochondrial DNA. Finally, so access to clinical trials remains predominantly limited to Western Europe and North America. We are currently missing a large population from understudied populations across the globe that would help power clinical trials. Things have improved recently, so as we can see in this figure here, so we have trials ongoing in most parts of the world, although Europe and North America remains major hubs for clinical trials. And designing clinical trials for rare diseases brings challenges. It's a balance between developing a trial for a rare disease, so trial power, control group, and developing biomarkers and outcome measures, with real-life population, so patients with comorbidities on multiple medications and that have normal life commitments. So sometimes we have on paper the perfect candidate for a clinical trial, but we cannot enroll them because of the comorbidities, the medications, or because they don't want to come multiple times for the trial visits. This slide summarizes the studies we have participated in. Some are complete and others are in the setup phase. As you can see, the majority are industry-led, but we also have one investigator-led study that is in this, we are still developing, that involves using the 2DG. There is a range of early and later phase studies which have adopted different primary endpoints and including different genotypes. But there is one overarching theme that is the focus on muscle-related symptoms, with in particular primary mitochondrial myopathies that are the target phenotype of the studies. So if we now talk a bit about biomarkers and outcome measures in mitochondrial diseases, we know that they are obviously pivotal for the development of clinical trials. What we want as the ideal biomarker is something that is obviously repeatable over time, that is cheap and ideally non-invasive. But for primary mitochondrial diseases, we don't really have approved biomarkers that we can currently use. And for outcome measures, this is something that helps doctors figure out if a treatment or an intervention is doing what it's supposed to do. So this is a list of the outcome measures that we are currently using in clinical trials for primary mitochondrial myopathies. You can see a variety of measures, and this is because of the clinical heterogeneity of these diseases, so we have to take into account different symptoms that these people have. But the sensitivity of these measures remain a big issue, and it's unlikely that modest drug effects will be detected during the lifetime of a trial using many of these readouts that are actually not specific for mitochondrial diseases, but most of these are used also for other neuromuscular conditions in general, like the 6-minute walk test, for example. I have recently done a systematic review of biomarkers in primary mitochondrial diseases, and this figure provides a graphical representation of the different biomarker areas in primary mitochondrial diseases. You can see that there is a variety of these, from fluid imaging, omics, histopathology, and lots of other types. Over time, so initially, as you can see, the focus was predominantly on traditional biomarkers, so mainly fluid and imaging, which account for most area of research in every year range. But in subsequent years, the scope of biomarker research has diversified. So there are some emerging areas, including multiomics, advanced imaging techniques, and novel genetic insights that have progressively gained prominence. And the number of articles focusing on biomarkers has steadily increased over the years, but there are some areas of priority, mainly the expansion of longitudinal studies. Only 5% of current studies are longitudinal. The remaining ones are all cross-sectional. There is a need of advancement of emerging technologies, like, for example, imaging modalities. And the biomarkers mainly have been focusing on diagnostic biomarkers. More than 65% of studies are on diagnostic biomarkers. But thanks to the genetic advances that we have seen today, we don't really need those biomarkers anymore. We just need to focus on prognostic and predictive biomarkers more. So all these to introduce the relevance of our muscle MRI studies and why we have done this. I will just introduce briefly two studies. One is longitudinal, is a quantitative MRI study and phosphorus spectroscopy study. And the second one is a new technique called oxygen extraction fraction MRI. So to capture not only the muscle weakness, but also other dynamic symptoms such as fatigue and exercise intolerance, we can use techniques such as the quantitative MRI, the phosphorus spectroscopy, and the oxygen extraction fraction. The first one is a static technique. The phosphorus and the oxygen extraction fraction are more dynamic techniques that we can record during exercise. So the first two techniques have already been tested in primary mitochondrial myopathies, but there are no longitudinal studies. There are no studies designed to mirror clinical trials. We don't know what are the correlations with outcome measures. Some of these studies show contradictory results, especially with regards to the phosphorus spectroscopy. And the oxygen extraction fraction has never been tested in primary mitochondrial myopathies. So the first study is the quantitative MRI and phosphorus spectroscopy study. So I have recruited 10 patients with 3to43 mutation and 8 patients with mitochondrial DNA single deletion with CPO. And I have performed an MRI at baseline and after 1 year follow-up. Apart from the MRI, I have collected clinician and patient-reported scales. You can see them in the slide. And measures of functional performance, including the 12-minute walk test, 30-second sit to stand, and some dynamometer testing. The quantitative MRI was targeting the thighs bilaterally, and the phosphorus spectroscopy, I've used the protocol developed by Slay et al. In Cambridge that includes putting on some personalized weight on the ankle of our participants and measure and do the acquisition at baseline during 1 minute exercise and during 4 minutes of rest. The exercise was a knee extension flexion. So now I'm going to show some of the most significant results. So at baseline, we have seen that there was an increase in the fat fraction. The fat fraction is the percentage of fat in the muscle of the thighs of our participants that was increased in the chronic progressive external ophthalmoplegia, so people with single deletion, compared to the 3243 patients that were behaving more like the healthy controls. When we were looking at what happens after 1 year, the fat fraction was increasing of 0.3%, which is not a lot compared to other neuromuscular conditions, but it's still a change that we are able to detect over 1 year with the MRI that we were not able to detect with the other scales, like the 12-minute walk test or the dynamometer parameters that were not showing any significant change over 1 year. Now, moving to the phosphorus spectroscopy results, this is just to show you what we were analyzing. So we were looking at a peak of phosphocreatine and inorganic phosphate pre-exercise. At the end of 1 minute exercise, the phosphocreatine was going down and the inorganic phosphate was increasing. And at the end of the recovery phase, after 4 minutes, the peak was exactly as it was pre-exercise. And we were able to measure two main variables. The phosphocreatine recovery time constant is measured in seconds. It tells us how fast the phosphocreatine is recovering after exercise. Obviously, we want it to be shorter because it means that the muscles are recovering quickly. And the second variable is the ratio of phosphocreatine and inorganic phosphate at rest. That tells us the energy state of the muscle, and a higher ratio is good because it means that there is a lot of phosphocreatine and the muscle is well energized at rest. We have seen that patients, both with the CPO and the 3243, were quite different compared to the healthy controls. They had a slower recovery time and a lower ratio compared to healthy controls. So their muscles took longer to recover after exercise, and it was less energized even at rest. And the phosphorus spectroscopy shows very good correlations with clinical data, as you can see in this slide. So it shows good correlations with clinical scales like the PMM, with subscales of the quality of life like the physical functioning or the pain. Correlates very well with the 12-minute walk test and the 30-second sit-to stand, and there was a positive correlation between the recovery time and the fat fraction, meaning that the more fat fraction we have, the more fat we have in the muscle, and the less oxidative muscles we have, and therefore we have a slower ATP resynthesis, which makes sense. There was no correlation with the FSS, that is the fatigue severity scale, and this is likely because the fatigue scales measure not only the muscle fatigue but also the brain fatigue, and not just the muscle problem. That's why they don't really correlate. So based on the main findings of these studies that are summarized here, I believe that a combination of the quantitative MRI and the phosphorus spectroscopy should be tested as part of clinical trials, especially the phosphorus spectroscopy. And all these measurements together can help stratifying patients with primary mitochondrial myopathy and enable a more precise enrollment for clinical trials. So, for example, we don't want to enroll people with an increased a high fat fraction, because obviously the fat fraction is not something that will resolve with the compounds we are testing. So we don't want to recruit those people. And the phosphorus spectroscopy would be good to check if the compound we are testing is actually having an effect on the muscles of our patients. Now I'm just showing a couple of slides about the oxygen extraction fraction study in muscle. This is because this study is still ongoing, and we are finishing the setup phase. So this is a relevant technique for primary mitochondrial disease. Because mitochondria utilize oxygen to produce energy, the amount of oxygen that is extracted from blood tells us how well the mitochondria are working. And we are able to measure that with the MRI because there is a different magnetic property of the hemoglobin compared to the deoxyhemoglobin. This is in summary our study design. So we acquire the oxygen extraction fraction at baseline, then we apply a cuff compression for 5 minutes on the thigh, and we measure the oxygen extraction fraction in the calves of our patient. Then we release the cuff and we measure what happens during the reactive phase for 5 minutes. These are the results of our 2 healthy subjects. So the stability test on the right is the resting state. People are just lying flat under the MRI, and we measure the oxygen extraction fraction. That is just a number. And we can see that it's quite stable over time. When we apply the challenge, so the cuff compression, the oxygen extraction fraction goes up, and then after 10 minutes, when we release the cuff, the oxygen extraction fraction goes down and go back to baseline. We are now discussing what would be the best parameters to compare to mitochondrial patients. These will be likely the resting state and the difference between baseline and the peak during the cuff compression. And we are planning to recruit the first person with mitochondrial disease quite soon. So we have seen how biomarkers and outcome measures can really help developing clinical trials. We have seen also how sensitive imaging biomarkers reveal what clinical scales sometimes miss and would probably enable smarter trials, although we are still at the beginning of this journey. So we will need now to test these techniques as part of a clinical trial. Thank you. That's the end of my presentation. I would like to thank the London Mitochondrial team and the Neuroradiological Academic Unit, and also all the funders. Thank you.
Thanks, Chiara. So we do actually have one more talk, and our originally planned speaker was delayed on a train, I think. So we have a slight change of lineup. So we're joined by Liz Curtis, who runs the Lilly Foundation, which is a patient charity that you've already heard about a lot today. We are really grateful to the Lilly Foundation for all the support they supply for our patients and our research. And she's going to talk a bit about a patient-centered approach to diagnosis, because we've noticed in the general genetics community with mainstreaming, sometimes the diagnostic experience can be quite negative for patients. And so the Lilly Foundation, in collaboration with the clinicians working in mitochondrial disease, have been trying to gather some hard data about this to talk about the experience of diagnosis, and hopefully we'll be working towards doing it better in the future. So thanks, Liz.
Thank you. Yeah, so I'm thrown into this quite last minute. Katie's actually in a taxi, so she might run through the door any minute and she can take over quite happily. So I've got her slides, very small. Fortunately, I brought my glasses. Bear with me. So yeah, I'm not Katie. So we're really proud today to be presenting and sharing the results of the first piece of Lilly Foundation-led research. It's a study that looks in depth at how patients and families experience the moment of receiving a diagnosis of mitochondrial disease.
It's an aspect of the diagnostic project that remains under-researched, particularly from the patient's perspective. And whilst most existing studies focus on caregivers, very few actually explore patients' own experiences of receiving a diagnosis. And through our work funding the National Precision Diagnostic Project that we've been talking about today, to improve diagnostic rates for mitochondrial disease, patient engagement work with our community has highlighted the need to look more closely at the quality of the diagnosis experience, not just at the science behind it. Oh, yeah, that looks right. Okay, so the Lilly Foundation, as you've heard, is funding the diagnostic medicine diagnostic project here, the national collaboration led by the UCL team and the mitochondrial centres across the UK. The project uses cutting-edge genetic techniques to help find confirmed diagnosis for more complex cases of mitochondrial disease that Renata has kindly explained to us this afternoon. Our involvement in this project has given us the opportunity to look at both sides of diagnostics, from the science to finding an answer and the human experience of how that answer is delivered. Through our Impact patient involvement group, volunteers with lived experience who shaped the Precision Medicine Project, they help shape the Precision Medicine Project. Patients and families have told us that improving diagnosis isn't just about speed and accuracy, but actually about the way in which these diagnoses are communicated. They describe a highly variable picture across the UK, with differences in who delivered the diagnosis, how it was shared, and where it took place. This has been echoed in what we've been hearing through our day-to-day work as a charity, with some families being told their diagnosis over the phone, by a non-specialist, or even by letter. It confirmed that while precision medicine is advancing the science, the human experience of receiving a diagnosis still needs attention. So alongside the precision medicine work, we partnered with Will and his colleagues here at UCL to explore this further. We commissioned the rare disease research partners to run an independent study guided by the priorities patients and families have already raised with us. The study aimed to do two things: to understand more about the experiences of people going through the diagnostic process, and to capture individual experiences of the moment a diagnosis is received. To explore these aims, we ran a national survey earlier this year to hear directly from the patients and the families about their experiences. The survey ran from April to May. It was open to adults with a confirmed genetic diagnosis of mitochondrial disease and to parents and caregivers, including those whose children had passed away. We shared it through our usual channels, by email, through our own private Facebook groups, and at patient events and specialist clinics. In total, 172 people responded. Of those, 166 gave consent and 103 met the criteria to be included. That's 103 people living with willing to share their direct lived experience of being told they or their loved ones had a mitochondrial disease. The living patients ranged in age from just over 6 months to 77 years, giving us a broad view of how experiences have changed and where they have not. With the survey complete, we moved on to the results, starting with what people told us about their experiences before receiving a diagnosis. Most people first sought medical help because of symptoms such as fatigue, vision, or coordination problems or weaknesses. For most, these were not isolated symptoms, but part of a pattern, things that developed or worsened over time. Some described a gradual decline, while others came to medical attention after a sudden deterioration. Adults often talked about long-term ongoing issues such as diabetes, hearing loss, or progressive weakness. In children, it was more often developmental concerns or acute problems such as not feeding well, high lactate, seizures, or regression. Altogether, it paints a picture of people navigating a complex, evolving set of symptoms long before they reached a specialist who recognised mitochondrial disease. On average, it took 9 years from first symptoms to confirmed diagnosis. For adults, the wait was around 5 times longer than for children. More than 15 years compared to just under 3. Nearly 8 in 10 people saw more than one specialist before getting a diagnosis, some as many as 11. More than a third were misdiagnosed along the way, and among adults that figure rose to over half. Several described feeling dismissed or disbelieved during this process, particularly before referral to a specialist centre. In our data, those who experienced misdiagnosis were often told their symptoms were psychological or attributed to more common conditions such as diabetes, fibromyalgia, or mental health issues. When it came to genetic testing, people's experiences were mixed. About 2/3 said that the process was clearly explained, and just over a third said they had support along the way. Many described long waits for referral, and only about 1 in 4 said overall experience was positive. That experience often came down to how the diagnosis was shared. Those told their results in person were much more likely to describe the process of as positive than those who were told remotely. Overall, 70% of participants received their diagnosis in person, usually in a clinic or outpatient appointment, sometimes on a hospital ward or in intensive care. The rest were told remotely, mostly by phone, some by letter, a few by video call, and 1 person through the NHS app. Over time, this has changed. In the 1990s and 2000s, 86% were diagnosed in person. In the last 15 years, that has dropped to 68%. So while genetic testing has become faster and more routine, it is now more common for results to reach patients and sometimes for professionals who do not have the specialist experience in mitochondrial disease. That can mean less explanation, fewer chances for families to ask questions or process what they've been told. The number of people who felt that the person delivering their diagnosis had experience in mitochondrial disease has also fallen, from around 3/4 to just over 40% today. Receiving a diagnosis of mitochondrial disease is life-changing. For many, it is also deeply distressing. 68% said the experience had a negative impact on their mental health, yet only 17% were referred for psychological support. That impact was most severe when news came as a shock or was delivered remotely. Among those who received their diagnosis by letter, 89% said it had harmed their mental well-being. More than half said they felt alone after diagnosis, and that feeling had become more common over time, raising from about 30% in earlier decades to 60% among those diagnosed in the last 15 years. Do you want to take over, Katie?
I just have a minute.
Okay, I'll carry on.
Sounds like you're doing really well, though.
Thanks. So, after diagnosis, experience continued to vary. Most received some written information, but those told remotely were much less likely to get a clear explanation or follow-up. None of those diagnosed by letter were referred for psychological support. Just over half were referred to a specialist clinic, and only a third were signposted to any patient organisation. Of those who were, 4 out of 5 named the Lilly Foundation, often describing that it was the first time that they'd felt supported. When asked what could be done differently, 6 themes stood out. First was the way in which diagnosis is delivered. Families wanted sensitivity, preparation, and ideally a face-to-face conversation. Second, faster and clearer pathways to testing. Third was a better understanding among healthcare professionals. Fourth was more attention to mental health. Fifth, diagnosis by the right specialist. And finally, being listened to. Taken together, our findings reveal a clear pattern. Long diagnostic delays, inconsistent delivery, and a lack of psychological support. While advances in genetics have shortened the scientific journey, the human experience remains uneven. Most people said that having a diagnosis was positive, bringing clarity and access to care, but the experience often left a lasting mark. And this is what we want to change. We're now beginning phase 2 of this work, a series of in-depth interviews with participants from the survey. These will help us to understand in more detail what good actually looks like and how to make that experience better for everybody involved. The goal is to publish recommendations for a gold standard in diagnosis, something consistent, compassionate, and informed by the experts. At the Lilly Foundation, we are already acting on what families have told us. Our new guide to Mito, created in partnership with Mito experts, is now being prepared for distribution across NHS clinics and around the UK, so no one leaves their first appointment empty-handed. Every one of the 103 people who took part in this study shared their story in the hope that by speaking up, things might change for themselves, their families, and others who will face diagnosis in the future. At moments like diagnosis, it is often the patient organisations who are there first, when families are frightened, confused, and searching for information. We see the human impact of how these conversations are handled, and that puts us in a strong position to drive change. This study shows that patient-driven research does not just describe the problem, it helps to shape the solution. Together with our partners at UCL and across the mitochondrial community, we're turning those voices into evidence and that evidence into action. At the Lilly Foundation, we believe that every diagnosis should be delivered with clarity, compassion, and hope, and that the patient voice should always be part of the science that makes that possible. Thank you.
Thank you so much, Liz, and for doing it last minute as well. This is a culprit if anyone wants to throw anything at it. So we just have a little bit of time now for some questions. I don't know if our speakers want to come back up. Does anyone have any questions in particular? Okay, so who's our roving mic? It's Áine. I might kick off then with a question for maybe Katie and Liz. So I think that this information is really powerful, and it also shows that this approach to letting anyone do a genetic test at any time isn't always completely helpful. Do you have much feedback from the patient communities in terms of what type of doctor or other specialist was kind of the most effective in giving the diagnosis, or that kind of more granular detail?
Yeah, thanks, Will. So I think what came across and comes across from just talking to families is that people expect and want a diagnosis from a specialist. Obviously, in more common conditions, you would expect the diagnosis to be given by someone who knows a bit about the condition. And what we hear quite a lot is people will get their diagnosis by accident, and might be a completely different specialist who has done a genetic testing panel, come up with the results, and then contacted the family to say, you have got this mutation, it's a mitochondrial disease, I don't know anything about it, we'll send you a letter in the post, you'll get to see a specialist in 6 to 9 months. And that's obviously not what we want.
Google it, though.
Oh yeah, that's the other thing, but don't Google it. Don't Google it, just sit and wait. And I think that's the experience you have a lot, Liz.
Yeah, definitely. It's exactly that. And you know, when you Google mitochondrial disease, it is a not very pleasant condition, and that is what happens. And we have very devastated, scared parents or patients who just think that their life is over, and it's very difficult. You've heard today how difficult it is to diagnose a patient and what everything means. So for us to try to make somebody feel better or safer, or it's incredibly difficult because we don't actually know what they're dealing with. So it's a real complex, difficult situation, and I think it's one that we face quite often.
Great, thank you both. And I think this data, if other patient support organisations were doing stuff like this as well, we would really be able to push back against some of the ways in which testing is planned around the country. So I think it's really powerful. So we had another question. Yeah, go ahead.
Yeah, hi, thank you. Really fantastic talks and fantastic day. My question was about the Lilly Foundation, about the psychological support in terms of if you're signposting a patient to the charity, what support's provided through you guys?
I'll do some work. So usually what we do at the Lilly Foundation, I'm talking for Liz now, is that so Liz continues to be the first port of call for any new patient diagnosed or going through the diagnostic process for mitochondrial disease. So Liz will always offer an initial chat, either a phone call or text or email, whatever. If after that initial conversation, Liz feels that this is a family who would be in need of some additional psychological support, or they ask for it, then we do have the partnership with a not-for-profit counselling service called Rare Minds, and those counsellors are specifically trained in the challenges of mental health in rare disease. So we have a partnership, I think it's 3 or 4 years old now, really successful partnership. We work with 2 counsellors who have got to know people with mitochondrial disease and some of the unique challenges. And so what we do is, it's always oversubscribed, but we tend to try to hold a waiting list of around 3 months. So what people would need to do, they'd need to self-refer. So either by email or conversation with Liz or myself, they then get put on the waiting list and then triaged from there.
Thank you.
You're welcome.
Thank you very much, Katie. I can also advocate for the Liz service as well, because we constantly have patients in the clinic who mention Liz by name and how good she has been to them and made a big difference. And you saw her in some of the videos that Bobby showed earlier about, doing stuff with the press and trying to get to make sure that the techniques like mitochondrial donation got over the line. So we're really indebted to this foundation and to Liz in particular. Is there a question up here?
I have a question for Renata, I think. So when you find these tandem mitochondrial DNA molecules or duplications, what kind of heteroplasmy level do you observe? And the second question is whether these findings which are pretty peculiar have been validated by other techniques like, I don't know, Southern blot maybe. Thank you.
So it is quite difficult with long read sequencing to estimate heteroplasmy, but we had patients in which there were far more deletion dimers than normal mitochondrial chromosome. And we had patients that had only maybe like a handful of these molecules that were deletion dimers. So it's very, very variable. And for duplications as well, now, whether we are going to validate it with another technique, I don't know. This is my go-to person for everything we may do next with whatever I find. So far we haven't validated it with anything, but I'm personally very puzzled by how variable the level of this deletion dimer is in very few patients that I have had with single deletion. So we need desperately more patients to try that.
Sorry, I feel like I was very rudely grabbing the mic. It's a favourite topic of mine. I'm really that cool. So in the clinic, we do next generation sequencing, obviously. And so for things like the duplications, we are inferring based on the orientation of the reads and stuff, and also knowing about the biology of mitochondrial DNA, that something is probably a duplication. And so we have that as a starting point. And then in some cases, we've worked with some people who've been working in this area for quite a while and have found some of these in historic samples using southern blots. But I absolutely agree with what Renata is saying about the heteroplasmy. It's kind of impossible for us, because what we're doing is we're selecting for certain reads in some ways. So we'll take out very short molecules. So are we missing something there? We could be, should we be looking only at super long reads, this kind of thing. So it's really hard to know exactly what the heteroplasmy level is, but we do think that everything that we find is real, but exactly what proportion it represents is tricky. Yeah. We've got a question from Bobby.
There's a question for Chiara. Variability is the hallmark of mitochondrial disease, and I was just looking at your data for the fat fraction change, and I think you said it was 0.3%. Was that a mean value? Because the data looked like there was some that clearly did change quite markedly, and others that just didn't seem to. And can you predict who it would?
Yeah, so it's true. So the overall change in the whole cohort was 0.3%. The main deterioration was driven by the CPO, caused by single deletion. The 3243 were not deteriorating that much. This is interesting because when we recruit primary mitochondrial myopathies, we recruit both, but they seem to behave differently. The number I have recruited is not enough to make more correlations, but yeah, it seems to be worse with age, and definitely the CPO are worse than the CPO are more similar to like classic myopathies. The 3243 are less, have more dynamic symptoms.
Great. Do you have any other questions? Oh, one more.
Thank you. It's a question for Liz or Katie. I think it really, your study really highlights the importance of sort of pre-test counselling, and it sort of highlights, I guess, the fallout of what that looks like when it hasn't gone well. But I guess, did you capture sort of what that pre-test counselling looked like amongst that cohort?
Thank you. So not really, but we did ask a question around during the kind of diagnostic process, was anything discussed with you around whether this could be a mitochondrial disease? So there was options of, it came as a complete shock, or there'd been some discussions, it was kind of likely, and the majority, I think it was mainly a shock, and there hadn't been proper counselling or even an assumption of that could be a possible cause, obviously in our sample of data. Yeah. Do you have anything?
Do I have anything to add to that? I feel like I'm really hogging the mic. Yeah, I think it's exactly as you said. It is quite surprising for a lot of people, but it is a function of who suggests the test. If you see a genetic counsellor, they are trained in what to say before sending the test. Whereas if you're seeing a very busy ophthalmologist, they'll barely mention in some circumstances that they're doing a genetic test, and that's not really a criticism of them. It's the way the NHS is structured at the moment, so it's quite tricky, I think. But I do think that people need to be warned in advance. So yeah.
With diabetes as well, we have received some referrals from endocrinology with women with 3243 diagnosed in the endocrinology clinic for diabetes, and they were not explained everything about that. They were just, okay, we are doing a bunch of genetic tests, your diabetes may be genetic, without saying anything to them. And yeah.
I think we have one more question up here.
So my question for Chiara, I really liked the oxygen extraction fraction. I have a question related to the methodology. So the patient, if we want to put a compression on the patient, so the other leg will be a control, or you are making the control a healthy patient?
So you are asking if
about when we put the compression on one of the leg.
Yeah.
So the other leg will be the control of this patient, or you are using control of a healthy population?
No. Controls will be healthy populations, okay. Because we want to see the main aim of the study would be to see how patients extract oxygen in a different way compared to healthy controls. So the other leg won't be tested. We are now during the setup phase, I didn't show the results, but we are doing some tests on both legs to make sure that they behave in the same way, and the results are that they are the same. But we don't have, otherwise the protocol will be too long, so we cannot test both legs during the same session.
Okay, so we've come to the end of the program. We do have some a networking reception just in the foyer outside. But before we move to that, I just wanted to re-emphasize this QR code to fill out the feedback. We really appreciate you filling that in, because that will help us shape this symposium for next year. And all feedback is helpful. I'd also just like to say a few thank yous. So I want to thank all the speakers for giving up their time. That's hugely appreciated, and all the people who've helped during the day with the roving mics and the AV, and in particular, although I'm not sure she's here, Adelina, who many of you have met outside, who really helped organise the day. So thanks to all of them. I suppose it'd be nice to give them a round of applause. I'd like to thank the Lilly Foundation for just being here, for contributing to the day with a talk, and just obviously all the support that they're giving us as clinicians. You've seen some of that today, the support they provide us, and obviously to all the patients and their carers. We'd like to thank UCB for sponsoring the day, and obviously we want to thank you all for being such an attentive audience. So thank you again.